You want to record or are you already recording? Okay. Gentlemen on the back. Okay, thank you very much for being here today. This is for SUNBIO diffusion, very important. SUNBIO is a project in which we develop intelligent ecological monitoring observatories that can be adaptively redeployed into areas of interest. So according to the European Biodiversity Agenda, this could be related in intersection with restoration. That's why Posidonia is a case of study for us, for mitigation and for promoting restoration. We are a group that comes from ecological monitoring and technology. So we have more than, I would say, 15, 20 years of collaboration with UPC within the framework of Technoterra unit, which is established for us, application scenarios in ecological monitoring, coastal areas to the deep sea, and then they develop the solution we need, okay? So thank you very much for being here. We would like to generate, let's say, a protocollar paper. I'm not an expert in Posidonia myself, you are the expert. So we will convey this information, we will gather the notes, and we will try to deliver a product. Thank you very much.
Yeah, a follow-up email will be sent to all the presenters, just a kind reminder.
Okay, then welcome all again. My name is Joaquín del Río. I will moderate the morning session of the Posidonia oceanica restoration workshop. Then welcome all of you, the ones that are online, the ones that are here. We have a quite tight agenda. Then there are a lot of presentations. We are very, very proud to be able to prepare such a kind of workshop with a lot of people that is very, very important on this topic. Then let's try to start, to be on time. Then I'm going to introduce Arianna Pansini. Arianna is a postdoc at the University of Sassari, Italy. Her studies focus on the Mediterranean seagrass foundation species Posidonia oceanica. Particularly, its project aims at testing the reliability and effectiveness of Posidonia oceanica restoration under an expected climate change scenario, regarding the effectiveness of past restoration actions, assessing the capability and resilience of seagrass to counteract or adapt the future environmental conditions, seagrass responses on restored sites. To do so, several approaches are used, investigating the seagrass morphometrical and biochemical responses, providing insights and perspectives that lead to efficient Posidonia oceanica restorations. Then I'm going to give the floor to Arianna. If you, Arianna, let me show your presentation. Can you share yours?
Thank you so much for the introduction. I'm sharing the screen. Are you able to see my screen?
Yes.
Yes.
Yes, we.
Perfect. So thank you again for the introduction. Today, I'm so happy. It's a pleasure to present as a first presenter. And today I'm going to show you the effectiveness of Posidonia oceanica restoration actions through a meta-analysis approach. Okay, so as I'm the first one, I am glad to present what Posidonia oceanica is, even if for the non-expert. For the expert ones, please apologize for the for introduce Posidonia oceanica again and again. But I would say that Posidonia oceanica is a seagrass, and as a seagrass meadows, it is one of the most important coastal ecosystems, providing worldwide ecosystem goods and services that contribute to the humankind. Okay. As other seagrasses, Posidonia oceanica is the iconic and widespread endemic seagrass of the Mediterranean Sea, but it is a very slow-growing species. As for other seagrasses, human activity and climate change are creating repercussions. Posidonia oceanica stability, resilience, and productivity. So, seagrass restoration could be considered as one of the conservation activities that can reverse Posidonia oceanica degradation. In fact, seagrass restoration is considered a process of reversing the degradation of ecosystems, improve the productivity and capacity of ecosystems to meet the needs for the society. This is from the UN Decade of Ecosystem Restoration. So today I'm glad to present results from a paper published in 2025, which aim was to provide a quantitative knowledge about the effectiveness of the Posidonia oceanica restoration actions. So the aim of the study was to understand the real effectiveness of Posidonia oceanica restoration action. Why? Because during the last 4 decades, so we are talking about 40 years of restoration, many seagrass restoration were performed along the Mediterranean Sea. So the aim was to collect this evidence and quantify the effectiveness. The study considered a research string on Scopus and Web of Science with keywords and Boolean terms referred to Posidonia oceanica, seagrass restoration, transplantation, and so on. And the screening process of the meta-analysis removed all the duplicate documents and included all the study that considered field trials, so not mesochasms, active restoration, so performed by the humans, and consider also the study in which the response variable were considered with a control group, a mean, a standard deviation, and a sample size, okay? Therefore, all the data was extracted by from 33 documents consisting of 1,223 case studies. How this study quantified the effect of a restoration? With a Hedges-G statistic effect. Okay, this is an effect size that considers a successful restoration action with a Hedges-G above 0 with a positive effect and negative effect with a values lower than 0. It was also considered a range of Posidonia oceanica restoration, which consider a small, medium, large, and very large effect in positive and negative, of course. So these slides are quite, I would say, with a lot of information. So I'm trying my best to reduce them. And for the quantitative and categorical variables that were included in this study, several variables were considered, from the physical characteristic of the study sites, the procedural restoration context, and the outcome. For example, for the physical characteristic of the study sites, this study considered all the characteristics referred to received site. For example, the presence of the previous presence of Posidonia oceanica in that site, the depth, the substrate, particular stressor, and so on. And for the procedural context, for example, several variables were considered, as the transplanted area, the density of shoot, the density of the transplantation, the anchoring technique, the beginning of the intervention, the monitoring period, and so on. And for the restoration outcome information, several variables were considered, as the type of the control group, that could be a reference method or the start of the intervention, several response variables, the mean of the transplanted group and the mean of the control group, the sample size, and the standard deviation. So just to localize the transplanting intervention, most of the transplanting interventions were performed in Western Mediterranean Sea, especially in Italy, France, and Spain. And these are the results for the quantitative variables referred to the physical characteristic of the study sites. We can see briefly that more successful outcomes were considered when the receiving site is shallow and when the distance from donor and the receiving side is very low, and when the donor depth is deeper than the receiving side. For the categorical variable, we can see that the restoration is unsuccessful when not considered the presence of a Posidonia oceanica, of course, the meadow substrate, the mechanical stress on the receiving side led to a positive success, and the absence of a stress on the other side led to a negative effect. So these results are probably linked to a poor habitat suitability when choosing a receiving site, a natural segress competition when transplanting inside a living meadow, the link to a past presence of a punctual stressor like the mechanical damage once it's removed can lead to a positive outcome, and the stress due to transplantation. For the procedural context of the transplanting intervention, the study identified a negative effect to the transplanted area. I mean, the larger the area, the lesser the success. Probably these results is correlated with a monitoring effect, since monitoring a large transplanting restoration can require lots of monitoring investments. Probably this is the reason why larger restoration led to unsuccessful outcomes. Regarding the plant, the higher the shoots inside the plant portion, the better the restoration success. Of course, the monitoring period, the more the transplantation is monitored, the better it is, and so on. For the categorical variables linked to the procedural context, negative effects were identified when considered MAT as an anchoring technique, transplanting during autumn, transplanting with sods, and transplanting during pilot transplantation, while positive outcomes were identified were considered rehabilitations, adding neuter inputs, transplanting with modular blocks, and plagiotropic rhizomes. So these results could be linked to a restricted monitoring area or a natural functional features of the plant, or maybe a better anchoring techniques in respect to other ones. For the restoration outcomes, of course, we can see that considering only the survival, the restoration cannot let a positive restoration success. But when considering the morphology of the plant, of the development of the plant, the restoration can be considered successful. So, of course, these results are linked to the feature of the explanatory variables. For example, the survival is binomial, okay? Dead or alive, or not considering higher ecological levels, okay? During this meta-analysis, the only response variables were linked to the plant itself. This is a very, very important result, because until this date, 2024, no higher ecological levels were considered for the restoration. So I tried to resume all these results in some panel plots. So again, it should be considered for the physical characteristic of the sites, the path of occurrence of Posidonia oceanica in the restoration site, because it is essential for the success of the restoration. Assessing the condition and environmental suitability after the cause of the decline, avoiding transplant inside meadows, and transplanting the diverse cuttings, guaranteeing the genetic variability. and should also be considered that moving cuttings from deeper meadows to the shallower ones to boost plant performance. For the procedural context of the transplanting intervention, it should be used plagiotropic cuttings, bearing at least 2 shoots. The use of drifted or harvested cutting to not affect the restoration outcome. Avoiding the transplantation during autumn, consider the additional nutrients during the first period of restoration. And for the outcomes, for the plant performance, it should be preferred also consider at first a small restoration area and then surveying for at least 20 months and then consider the whole restoration area. Investigating, of course, more than one plant trait to define plant performance, not limiting only on survival, and estimating higher biological and ecological levels as ecosystem structure and functioning for Posidonia oceanica. So I would thank you, and I will best all my wishes for your research. Thank you.
Thank you very much, Ariana.
Thank you, Ariana. Because of the time, and we are just keeping the time, there is an online link for the Slido environment where you can put your questions. Ariana, we can use also the link there to answer the questions, and then if we have a chance, then we can discuss a little bit. Now let's go to the second presentation that will be under Luca Rindi. Luca is a marine ecologist at the Department of Biology, University of Pisa, and his research focuses on the stability, resilience, and regime shift dynamics of coastal marine ecosystems, with particular attention to foundation species, climate-driven stressors, and biodiversity change. He works on the development and testing of early warning indicators to support a more predictive approach to marine monitoring. At the SUNBIO Workshop, he will contribute his perspective on the role of field experiments in providing key insights to support the restoration of Posidonia oceanica. Then, Luca, the floor is yours. Thank you.
Thank you so much. And thank you. I'm glad to be here and to present the work of our lab. We are all experimental ecology, so today I'm going to talk to you about the challenging opportunities linked to restoration of Posidonia oceanica, and in particular, how ecological experiment can help and improve restoration practice. First of all, I will skip the presentation of Posidonia since Ariana has already been going through a lot. So I will just directly present the main idea behind this talk, that basically restoration involves a sequence of practical questions and choices. First of all, one is where to restore. So the recipient site suitability, that is a key point to take into account. Not all sites are equally suitable, and local environmental condition, residual stressor, and other hydrodynamic condition can influence where, for example, the donor material can survive and establish. A second key point is where to collect donor materials. So also the plant material from different origins may differ in condition, growth potential, and also tolerant to stress. Therefore, donor choice is not simple a logistical decision, but also an ecological one. So we need also to consider how to handle and transport the donor material from one site, the donor one, to the recipient one. So to take into account the stress in the dislodgement and the translocation. And this is a key point that can influence and determine the success of the restoration initiative. So the goal here is to move from, to use this information and to answer this question through ecological experiment. For example, a key ecological experiment is reciprocal transplant experiment that has been historically used to disentangle, for example, the contribution of genetics versus the environment. And this experiment, this question lies and in this, for example, in this experiment, as tightly linked to the restoration ones that have been through already. So here is to use this experiment to not just to highlight and understand ecological mechanisms, but also to provide operational information for the restoration of Posidonia oceanica. So within this talk, I will present two main and interconnected activities. The first one is a restoration trial that we established and started in Geltrú Island in Tuscany Archipelago. It's a pilot action to test and assess the feasibility of Posidonia oceanica transplantation under field and restoration conditions. The second one that is guided from the first one, so this restoration initiative, is a repeat transplant experiment to disentangle the contribution on the restoration success of the origin of the restoration material, the recipient site, and also the handling and the stress involvement in the restoration, in the movement and transport of the restoration materials. Just to briefly present the Tuscany Archipelago, it includes 7 islands and that differ mainly between different levels of management and protection. We have fully protected islands like Monte Cristo and Pianosa, down to unprotected islands like Elba and Giglio, and in the middle we have Giannutri and Capraia that are partially protected islands. The economy and tourism is the main economical driver, and this attract a lot of tourists. And the number of tourists has skyrocketed in the last years, and this also increased and exposed the coastal area to both activity and, of course, anchoring. And this anchoring, of course, affected the main coastal habitats that are mainly in Tuscan archipelago are macroalgal forests and, of course, Posidonia oceanica meadow. This restoration initiative took place in Giannutri Island in 2 sites and starting summer 2023 and was a part of the European project Marine Savers. First of all, we involved the transplantation of fragments collected from a disturbed area that is currently exposed to anchoring to the boat activity to a disturbed area here that, as you can see here, show transplanting a dead material next to a meadow that show clear sign of previous exposition to anchoring and likely due to that occurred before the establishment of the marine protected area in Giannutri. So we use our transplantation. The transplantation technique that we used was to attach cuttings and so fragment plagiotopic rhizome with at least 3 to 4 vertical shoots to a U-shaped iron rod and put it into this dead material. These basically are our restoration unit. And we choose the restoration technique, transplantation technique, because it's, according to the literature, provide a nice survivorship on a mid-term, and at the same time was easy to implement. Here just to a scheme showing the restoration, how we restore. Each restoration unit was planned into a restoration path composed of 16 restoration units that were allocated in a range in 50 restoration patches or 1 meter square, 2 meters apart from each other. The restoration success was assessed using multiple response variables. We measure the shoot density, live shoots, number of live shoots per patch, the photosynthetic efficiency and the light adaptation using underwater fluorimeter, the leaf morphology, and the daily leaf production using the puncture method. So coming to the results and to the crispy part of the restoration, just to show you the shoot density across time. This is the time 0, the 2023. And we didn't observe much of an increase of a change in the restoration, in the shoot density in the next year. But in 2025, last year, we observed an around of 25% increase in shoot density. Regarding photosynthetic efficiency, we didn't show much of a difference between control and transplant sites. In terms of leaf morphology, number of leaf didn't show any particular difference between control and transplant site across time, but instead the total leaf tissue was significantly lower between control and transplant in both years, although in 2025 we observed a slight increase in total leaf tissue. And this was accompanied at the same time with an increase in brown tissue, necrotic tissue, but we didn't observe an increase in the leaf elongation rate, a change in the leaf between transplant sites. Basically, these plants got smaller, and this probably was due to, we think, an allocation of metabolism that were directed to the development of roots. So this transplantation and restoration initiative guided the second step. We wonder, since we are marine experimental ecologists, wonder which factor determine the success or the failure of Posidonia transplantation, and how does cuttings manipulation affect transplantation outcome and success. So first of all, we identify the factor affecting the success of restoration, the stress related to transplantation, so the dislodgement, so removing the cuttings from the middle, the detachment, and also the transportation, detachment plus the stress involved in moving the cuttings from one side to another. And then another key point, as I said, is the origin sites and the destination site. For this reason, we established a reciprocal transplant experiment to disentangle this effect, the contribution of this effect, in two sites in the same area, from Punta Secca and Cala Brigantina in Geltrú Island. The experiment involved different conditions. The first one that we established was the translocation one. So we detached the rhizome and moved few meters in the dead material next to the origin area. So, and then we had the transplantation plus stress that is similar to the previous one, with the addition that these cuttings and rhizome were brought outside the water and maintained in seawater for 2 hours to simulate mimicking the transportation from one side to another. And finally, we had the reciprocal transplantation, so moving the rhizome from one side to another and vice versa. And of course, we have the controls representing the middle of the two sides. The restoration cycle, I mean, the response variable assessed to see and assess the outcome of this experiment were the same that were being used for the restoration initiative, and that were evaluated after 1 year the setup of experiment, so last year. Just to show you the shoot density, as you can see here, we observed two interesting things. The first relies on the effect of the origin in interaction with treatments. We didn't observe a difference among treatment and interaction. So the shoot density in Cala Brigantina was significantly higher, but when we had the stress than the one in Punta Secca, that for the translocation, well, when we had stress due to the transportation through translocation plus stress, this difference decreases. So also the origin matters and can modify and the translocation effect. But another important thing is that also the transplant, although this difference between these two conditions, the transplanted one was not different, was not significant, you observe a tendency in terms of shoot density of the transplants horizon to assume and tend to shift towards the one in the destination, the recipient site, and vice versa. This pattern was also observed for a similar pattern, as you can see, was observed also for the leaf surface area, in which tend to switch. Again, when transplanted in a new site, the leaf surface area tend to become to decrease for the one origin from Cala Brigantina and move to Punta Seca, and vice versa for the one move from Punta Seca to Cala Brigantina. And this part was also observed for leaf elongation rate. So try to summarize and to conclude the results from this presentation is that I like both the potential, the complexity, this presentation and these two parts of the presentation show both the complexity and the opportunities to use ecological experiment. From the ecological point of view, our result show that transplant performance could be strongly dependent on the recipient's site. Plants do not respond only according to their origin. Local condition at the restoration site can strongly influence the shoot density and leaf performance. At the same time, donor still matters. I mean, it's not just the recipient site in relation to the translocation and transport related to stress. So the translocation and related stress can be modified and influenced by the origin. This means that donor material should not be considered fully interchangeable because different origin may respond different to restoration process. However, this is not also a methodological challenge. Restoration experiments are not easy to implement. They are very time demanding. They need proper replication. In fact, we have just 2 sites. A properly replicated experiment would have required more than 2 sites to assess this, but can be used to assess the restoration bottleneck and identify restoration bottleneck for specific sites. So this is just the take-home message that I would like to present, introduce, that can be used, this experiment for specific area, tailored for specific area, to identify potential and bottlenecks of future restoration initiative. So, and thank you for your attention.
Hey, Luca, thank you very much.
Yes. Yeah, thank you, Luca. Let's go for the next one.
One question.
Yes.
If we have a question, we were going to put it in somewhere.
Yeah, there is an online link.
But
There is on the Zoom.
Inside the Zoom.
Yeah, inside the
But
Zoom.
just in case, you can keep your question to the networking session
Okay.
and ask. You don't have to wait.
Okay.
Yeah, time.
Okay.
Yeah, then let's go for the third online presentation. Then we will move for the first in-person presentation. Then the next one is for Fabio Blanco, that started his career at the University of Alicante, focusing on the ecological impacts of desalination plants on benthic assemblies. Among those, Sigras caught his attention and passion, and his research has focused mostly on them since then, from gene expression to long-term microscale variation. His current research at the Stazione Zoologica Anton Dorn at Italy focused on the application of novel techniques such as assisted migration and stress hardening to improve Posidonia oceanica seabed-based restoration. Then Fabio, floor is yours.
We kindly ask Luca to stop sharing his screen.
Yeah.
Please, Luca, if you hear us.
Sorry, and I have a question. For the networking session, are the people that are online also going to be present?
Mm-hmm.
Okay, cool.
Okay, good morning, everyone. Can you hear me and can you see my slides?
Yes, thank you.
Okay, perfect. Okay, I think you might be, okay, are you seeing the Zoom window or just the presentation?
the presentation with the pointer, yeah.
Okay, perfect. Well, good morning, everyone. Thank you, Arianna, and thank you, Luca, for your presentations, because it already gave a little bit of background on seagrasses and on Posidonia restoration. And my presentation is called Does Origin Matter? And exploring assisted migration techniques for Posidonia oceanica restoration. This presentation and this work is inside the Seacovery project, a project that started in the Estación Etológica Antón Dorn in collaboration with the Spanish Institute of Oceanography, and which aims to use different biotechnological tools for assisting seagrass natural recovery. And we are talking today about specific objective 1, which is evaluating and testing assisted gene flow to promote seagrass recovery. It's important to say that this project aims to work on Posidonia oceanica seedlings instead of adult plants. So first of all, let's talk about what assisted migration or assisted gene flow is, and is the aiming to use local adaptation, that is, for example, the tolerance of certain genotypes to, for example, an stressor such as temperature, and try to use that local adaptation in our favor. So, for example, a classic example would be taking genotypes or taking plants that we know are subjected to higher temperatures and moving them to areas that we expect to have higher temperatures or are already having them right now, in order to have not only successful restoration now, but successful and resilient meadows in the future. And we have a lot of proofs and a lot of evidence that there is in Posidonia oceanica certain adaptations to, for example, temperature that we might take to our advantage. However, system migration have some risk, although they haven't been assessed almost in the marine environment, but in terrestrial systems, there are several cases in which moving genotypes from one place to the other not always have the positive impact that we expect. So that is something we have to study and consider before using this as a technique for Posidonia oceanica restoration. So first of all, before talking about gene flows, you have to know how are the genes distributed in for the species you are studying. And now we have a lot of information in Posidonia oceanica, although some areas are lacking. But we know, for example, that Posidonia oceanica, as it's an endemic species in the Mediterranean, has a very strong genetic isolation between western and eastern basins. So there is no genetic connectivity, for example, between plants in Greece and plants in Spain, not naturally at least. And then we have two areas around the Sicily Channel in which there is a little bit of mixing. Of course, this is not like there's not a strict barrier, but we can say, okay, there are some limits on how genotypes can move, and we should, just for precaution, keep those limits aware. If a gene will not move naturally from one area to the other, we should not play with that, but move in our bioregion and try to, if we do assisted migration, always do it in the areas in which natural connectivity is possible, at least. That's very important. So our question was, could we use heat-resistant genotypes to enhance restoration success and future meadows resilience? Okay, let's try to answer this. So what we did, we collected seeds from 4 different locations, 2 in Campania near the city of Naples, 1 in the same region, but in a southern area, and 1 outside of the region, within, of course, the genetic connectivity area in the south of Sicily. So we expect these Sicilian seeds although they are not exactly from the place you collect them, they are from the around area, to be maybe more resilient for warmer and warmer Mediterranean conditions, even in the Naples region. We transplanted them in November after 6 months of growing them in mesocosm, and then we monitored them in January and March this year. We also collected samples for genotyping. That's what we are still doing. And after the summer, we will study how pigments, antioxidants, and target genes are responding in the different plants from the different origins to this new transplantation site. These are very, very preliminary results. It's only 2 monitoring sampling times, but right now here on the left, you can see the nearest plants, which are from the island of Ischia, whereas on the right in purple, you can see the Sicilian ones. In terms of growth, although Sicilian performed better than the regionals in Nerano or Cilento, they did not, let's say, win the native ones. They produced more leaves, that's true, but then their survival was lower, was the lowest between the transplanted populations. So, okay, these are very, very preliminary results, but at least what we can see is that there is not a very clear advantage for the Sicilian seedlings to thrive in Neapolitan waters. So, first of all, we saw that growth and survival follow usually opposite trends. The ones that survive the most are not the ones that grow more, but that's a strategy that the certain populations might have. Makes sense. We saw that plants from the closest location to the transplantation grew more and had fairly good survival data. So, okay, first of all, we should support still the local genotypes. And assisted migration by now does not appear to have a very clear advantage compared to local populations. Okay, that's fine. We have to see what happens in the summer, in which temperature will be higher. But then we came with another question. Even if they don't behave a lot better than the locals, could they become like the areas of high production of seeds, such it happens in the Sicilian coasts, a source of material in case of low local seed production? So in case we don't have seeds to make restoration, is it a good idea to take them from other areas? That's something we have to answer. Also, because, for example, here in Spain, since February, every mitigation measure done by any kind of administration to compensate loss in Posidonia oceanica has to be done with seedlings, not with adult plants, and that's by law. So there's going to be much more work in seedling transplantations. We have still in the sea a lack of regulation in the guidelines or limits for the genetic resources translocation, whereas in the terrestrial systems, everything is very clear. We know if we are working with forests or trees, there is very clear where you can take the genetic resources and where can you put them. There's a lot of studies being done, not in the sea, so we have a lack of knowledge there. And in any case, we have to do genetic connectivity assessments, being sure that the genes we are moving would naturally arrive there somehow. We are not taking something, we are not breaking the genetic isolation. So could we use these areas of high recruitment for restoration material that happens in the Balearic Islands in Spain, that happens in Sicily, areas in which seed production is much higher, is much common. We know that it's not regular, but in these areas it's much more. Is this something we can do? We should do? We should plan? That's an open question. Still, we need very much detailed genetic connectivity between the areas. We have to reach a scientific consensus. It's not a yes or a no. We have to talk about limits. We have to develop guidelines. This is work we still have to do. And of course, that cannot go against the preservation and protection of the natural population. Even if it looks like an advantage to do these assisted migration techniques, that should not go against preserving local genotypes. And just this week, the recommendations from the European Seagrass Restoration Alliance came out, and part of these conclusions and these thoughts that I'm sharing with you are also written there. So if you don't have already done it, have a look at it, and always consider genes when moving plants. And of course, I'm just presenting this, but this is the work of a lot of fantastic people collaborating from Lázaro Marín here in Spain, and all the group of Gabriele Procaccini in the Stazione Zoologica, together with the El Parco Sommerso di Gaiola, a marine protected area in the near Naples. So of course, thank you to every single one of them, and thank you all for your attention.
Yeah, thank you, Fabio. Great. Let's go to move. Yeah, okay. In person presentation, you have this. It's on the stick or?
No, it's on.
It's on the stick. Okay, let's move on to the next presentation that will be on Craig, that is a marine ecologist and biostatistician with extensive experience in fish ecology, habitat structure, and applied marine science. He has worked across academia, government, and industry in Australia, California, and Europe, specializing in interdisciplinary marine and oceanographic research. More recently, his work has focused on fisheries technology, ecological analytics, and blue carbon systems. He is currently Chief Science Officer of Seagrass Blue.
Thank you, and thank you very much for the invitation to come and present. Yes, I am the Chief Science Officer of Seagrass Blue. It's based in France and UK. It's a bit of a sort of noble title because I'm the only science officer in the company. My background is in marine ecology, experimental marine ecology, in particular habitat, fish association, to really getting into the water and trying to manipulate systems and see how they function. What I'd like to talk about today is work I'm doing with Seagrass Blue, which is looking at whether we can develop midwater pods to cultivate seagrass, various species, in this case of Posidonia, in order to assist restoration projects. Right. Posidonia is sensitive to a wide range of stresses and threats, and one of the mitigation procedures is doing restoration. Okay, sort of as a last ditch effort in place of preserving and preventing damage. And the literature is pretty consistent in the things that go wrong with these sort of efforts. As soon as you plant something out into a potentially hostile environment, you get hydrodynamic stresses. This is often associated with sediment movement, which is not good for a plant that's trying to anchor into the seafloor. If they did become established, you have freshwater herbivory, it gets crabs eating your seeds or digging up your plants. And this combination of environmental variability leads to highly variable success rates in establishment of Posidonia. Okay, so this brings us to what Seagrass Blue does. What we did was we thought, let's have a look at another way of approaching the problem. Let's see if we can develop a tool that might help in some situations. So we thought about doing a bit of a conceptual shift. So typically we would go out and we'd plant Posidonia for restoration where it needs to be, which obviously makes sense. But the problem is these new propagules end up in this potentially hostile environment. So what we thought is, well, what if we could introduce an intermediary stage where instead of just planting them straight out where they're still young and tender, can we put an intermediate aquaculture stage in the water, so in the natural environment, in the real world, in some sort of platform that grows them in mid-water and try and remove some of these stresses? So that's essentially what we do. Now let's have a closer look at how that could work. So we could construct a pod that floats at the depth that we want, fill it up with the sediment or the substratum that we want, plant our seeds, however method we choose, whether it's rhizomes, seeds, seedlings, and then hopefully it'll grow and establish. A couple of things to note here is that with the floating platform, you're not restricted to deploying it at the depth at which the seagrass is going to end up. And we could moor it in 20, 30, 40 meters, as long as the platform's at the depth we want. So we have the ability to control the light and the temperature regime by setting the platform height. Hopefully, if the platform is constructed properly, then we can reduce some of this sediment movement, which is a big problem with that early root establishment. So in this way, we're sort of trying to take control of some of these factors, some of these environmental factors that are responsible for this variability in success. Being a platform is something that we can control means that we can treat it like an experiment. So we can treat it as modular blocks. We can accumulate them into larger sort of areas if we want to do this at scale. And we can deploy integrated monitoring systems. For example, environmental sensors to tell us what the light, the temperature, the salinity is doing. Time-lapse video to measure growth, measure biodiversity as one of these extra benefits of growing seagrass, and work out if something's going wrong. If you've got herds of groups of herbivores going through and trashing everything, then maybe you can adaptively respond to what's happening. And if these instruments are linked up to transmitters, you can get real-time insights. So you can have things going back to a data dashboard, back on land, up in the cloud, and you can see exactly what's happening at your various systems. And this is really what Seagrass Blue is about. All right, we were fortunate enough to get a modest grant from EMSO Eric to deploy a pod as a proof of concept at the UPC Observatory just off of Vilanova. This was an ideal place for us because, first of all, it gave us a site to work in. Whenever you're putting some sort of structure in the water, you need some permits. The UPC Observatory had that all wired for us. It's located 4 kilometers offshore and about 20 meters of depth, and it's protected from bottom fishing activities. So here we have sort of like a little outdoor marine lab in which we can deploy our system. There's also this infrastructure associated with the observatory. So we have monitoring access, like Kevin and his crew periodically go out and have a look at what's happening. There are communications lines. We have 4G. So we have all of these things that we can do to develop the technology that's associated with the pods. And it also provides us a good opportunity for collaboration with UPC, from the Restoration Ecology Group and the engineers and just the basic the observatory operations. So this enabled us to put out our proof of concept pod and real conditions, hopefully to get some real insights into how it works. Okay, the deployment itself was a fairly standard pipeline of engineering. We constructed our test pod, which is made from timber. It was actually constructed by the other co-founder of Seagrass Blue in his garage in France. We transplanted it down, we assembled it at Vilanova. It's, as I said, timber construction with mussel floats to keep it positively buoyant, about 12.5 square meters, so just small, just enough to get us a proof of concept going. We used a local marine contractor to deploy it because the systems are heavy, particularly with mooring blocks. It was deployed where we anchored and stabilized and made sure the thing was doing at the right level, we're doing what we wanted it to do. And then we planted it with seagrass plants. These were derived from beach rack, and they were being held in the aquarium at UPC by their crew there. So they took care of that sort of permitting. And then finally, after we planted, we had pulled our mooring instruments out, our ocean sensors plus the video. Okay, so. Okay, let's talk about some of the early results. So this was really a proof of concept, not a hard sort of data-driven exercise. My other partner is based in France. I'm based in the UK, so we couldn't get back to monitor as much as we want. However, what we do know is that plants survived. They didn't all immediately fall over as soon as we put them out. The video that we periodically got, courtesy of Joaquin's team, showed that, yes, the plants were okay. The last sort of video, there was a little bit of algae, a little bit of sediment accumulation on some of the leaves, the sort of seasonal growth that you get. The pod survived its first winter, so we put it out. It got deployed in November, and during that winter there was 8-meter swells, and the thing was intact. So it remained operational. It didn't flip upside down or tip up or anything like that, which was heartening. There was some sediment loss that occurred. So these are the sorts of things that we file away for future reference. Okay, what we don't know yet, this is one of the problems of working in the field in the real world, is at the moment we cannot find the pod plus its 2-ton mooring block. Joaquin and myself were out looking for it with a fishing boat with multi-beam sonar. It's just not there. And the current hypothesis is the fishing skipper thinks that maybe a fisherman found it and has taken it and put it somewhere as his personal fish aggregation device. So that's our current hypothesis on where it might be, which surprised both Joaquin and myself. The pod disappearing, yes. The big concrete block it's attached to, no. So we will continue to search for it and take the best guess. It might be a drop in the ocean. So what we don't know yet is its current location and the longer-term survival of the plants and the longer-term sediment retention. However, for a proof of concept with a very basic construction and very sort of rough mooring system, the system is showing potential. So our key lessons, okay, what worked? The deployment pipeline worked. We can construct, we can deploy, we can get it where we want it in the ocean. That is good. It is survivable. We are monitoring integration, our instruments, that all seem to work, although there's still a bit of tweaking there because we haven't got our live feeds ready yet. But that's still work in progress in other places. And the transplants persisted for the duration over which we could get video. What needs refinement is we need to sort of alter our design to make it a bit more robust, a bit more less likely to disintegrate with shipworm after 4 years. Change our mooring type to make it less impactful and work on the sediment retention. However, at the moment, we're looking like we have some sort of operational viability. Okay, there's no point in having a proof of concept without actually taking it further to something that's actually going to be useful for seagrass restoration. So we have a set of development priorities as our next stages. These are broadly engineering and ecological. From the engineering point of view, we need to work on different mooring systems that can withstand storms. So we need to look at loading and buffering the system. Sediment retention we need to closely look at, because if waves are going to either bounce the sediment out or push up into big dunes, we need to alter that some way. So we're looking at substrate and some sort of other baffling systems, maybe potato mesh. And as I said, the platform design. One of the things we're working on is having a platform that can change depth. So if we have thermal events coming through, which we see from the instrumentation, we can get the platform to, say, go below that temperature depth. Or if we want to stress plants before they're outplanted, maybe we can do things like that. So we're trying to make the platform smarter. Ecologically, what we want to do is expand this at multiple sites. We do have applications in to work with different species, different areas, from Denmark through to the Mediterranean, and really try and reduce this effect of the environmental variability on these early stages of Posidonia establishment. But ultimately, this comes down to the ecological validation. Can we generate sort of a floating source of material that can be used for restoration? Is it going to be cost effective? It won't be applicable in all cases because we have a huge Posidonia bed just there. There's no point in putting a pod. But if we have difficulties in getting the plants established, or there's a restriction with source material, maybe this could be one of the solutions that we have available to us as restorers. So the next challenge really is a combination of scaling and reducing these uncertainties. So as a quick summary, Posidonia restoration has this bottleneck of trying to get trying to get the roots established, trying to get that beginning of that map so that the plants can actually take hold. Posidonia is such a slow-growing species; it's a real pain on the backside. We think that one of the tools we could have in the arsenal is midwater establishment, midwater aquaculture, to try and get past that variability. It's not going to be the solution for every case, but it's just one of the things that we might have a weapon in the arsenal. One of the things that was intriguing for me as an ecologist is bringing in the engineering side of things. Chris Lewis, my other partner, he's a technology geek, so he invents all of this stuff on the ecology side of things. So working with the monitoring systems, working with the marine engineers, working with the sort of the mooring aspects, that sort of thing is actually quite intriguing. And maybe for ecological restoration, if things aren't quite working, maybe that's one of the things you can do is look outside the box and look at, hey, what can we engineer in the environment to make the solution work? So that's one thing I thought was kind of interesting about the current status of the project. So I'll finish just with a few acknowledgements. The funding was EMSO-ERIC physical access. The UPC Observatory was invaluable. Joaquin has been an absolute legend in getting everything organised and sorted and done for us. As I said, Chris is up in France, I'm in the UK, and Frederick kindly donated the plants that we planted out as part of our proof of concept. I'll end there, and I guess we take questions later.
Okay. Great. Thank you, Rich, for the question. Let's discuss a little bit. Okay, then let's move on to the next one. That is for Arnaud Bollinger. That is, he is a postdoctoral researcher at the Stareso Research Station in northwestern Corsica, where he has been working since 2021. He completed his PhD at Laboratory of Biological Oceanography at the University of Liege in Belgium, focusing on the restoration ecology of Posidonia oceanica. His current research explores the upscaling of restoration strategies by interacting facilitation processes alongside assessing the recovery of ecological functions following large-scale transplantation efforts. Then it's ready. Then the floor is yours, Arnaud.
Hi, good morning. Can you hear me?
Yes.
Yes, we can.
Okay, perfect. Thank you. First, thank you for inviting me to present this work performed by, as you can see, many different partners. So the short presentation of today, in a nutshell, will be on the multi-scale assessment of the biological and ecological responses of Posidonia oceanica to transplantation for the optimization of seagrass meadow restoration. So first of all, a bit of context. Well, there are some key questions that need to be asked before performing any seagrass restoration project. Basically, where to plant, what to plant, how to plant, and why to plant, which are some of the questions that have been partially answered during the previous presentation. During this one, I will focus on those two main ones, what and how to plant. First, what to plant. There are three main kinds of biological materials that can be transplanted, basically, either sods, seeds, or cuttings, cuttings being the most common approach. But there's also the question of which kind of cutting should be used in restoration projects. In fact, there are two main kinds of cuttings, either what we called donor meadow cutting, so that manually extracted from reference pristine meadow, in the case here on the natural sandy intermat, or you have what is called storm fragments, so naturally detached Posidonia rhizomes that can be detached by or natural event like storms or intense hydrodynamic activity, or human activities like anchoring. Then the second question is how to plant. How to plant, there has been many different trials performed for the last 40 years all over the Mediterranean, testing different methods, but there have been very little experiments comparing under the same environmental conditions, different methods. So in the experiment we performed in Corsica and Calvi Bay, we tested 3 different methods with different structural complexity, but also with different costs. The first method is called iron staple, as we've seen also in previous presentation. Second transplantation method is a coconut fiber mat. And the last one is what is called base element. So it's a biopolymer made of potato starch. So as you can see, there is a gradient of structural complexity with almost none for the iron staple and a high 3D structural complexity for the base element. And also the associated cost is quite different. The objective were the two main ones. First of all, do the stone fragments perform as well as the no-meadow cuttings in transplantation projects? And a second main objective was which of those three transplantation methods perform best and under which environmental condition. To answer those two main questions, we did a transplantation experiment in Calvi Bay, so northwestern Corsica, where we had 7 experimental sites on dead mats at two main depths, 20 and 28 meter depths. All the transplantation were performed in spring 2022, so 4 years ago. There's a total of 693 Posidonia fragments transplanted. Each time for each of the 7 transplantation sites, the 3 methods and the 2 donor sources were tested. And regarding the donor material, it was always a plagiotropic rhizome of at least 50 centimeters long, 3 shoulder shoots, and also very important to note, all of the cuttings had almost no roots. And then as it's classically done in all kind of restoration projects, we looked at the survival of those 693 cuttings. And what we found out is, well, first of all, for the base elements of the biopolymer of potato starch, as you can see in purple, it's almost the same survival rate either for shallow, so 20 meters, and deep, 28 meters. The coconut fiber mats performed poorly, 31% in shallow sites and 54% in deep sites. And what is particularly interesting for the iron staples is a not so good survival rate on shallow sites, while on the deep sides is a very good survival rate with 83% survival 3 years after transplantation. And the main explanation we have to explain those different behavior because of the depths is due to the hydrodynamic activity, which can still be quite intense on 20-meter depths, while it's almost absent for the deep sides. And as the iron staples do not provide any structural complexity and therefore any protection against hydrodynamic stress, it was the main hypothesis to explain those results. Regarding the difference between, so what do we call the intermat cutting, so the one extracted from donor meadow and the stone fragments, there were no significant differences in survival 3 years after transplantation. We looked a bit deeper, and the second trait we looked at after survival was morphology. And during this presentation, I'd like to focus on the roots, which is very little done in transplantation project. And as you can see first on this picture, and what we notice is that for the base element on your left and the coconut fiber mats, well, as you can see, 2 years after transplantation, there are almost no roots. 2 years and 3 years after transplantation, it was the same observation. While if you take a transplanted by means of an iron staple, as you can see, well, the root system kind of look like a natural Posidonia oceanica rhizome in a pristine meadow. We saw first that visually, and we decided to quantify it. So we measure a big number of different root morphological traits, the number of primary roots, the rooting depth, the maximum horizontal spread, the number of lateral roots, and it was a similar conclusion for all of those morphological traits. Here's the result for the total root length ratio. And what we found out is that for a control meadow, so natural reference pristine meadow, there was, well, a very well developed root system, significantly much more developed than any kind of transplant. And then second main result is for the iron staple, which has a significantly much more developed root system compared to both coconut fiber mats and base elements, which have basically no roots, even 3 years after being transplanted in the field. And then we checked again between our 2 donor sources, and there were no main significant differences in any of the root morphological traits. which leads us to the question of what's happening inside these roots. And then we thought about what is done commonly in terrestrial ecology in general, either forest ecology or even agriculture. Often the root system is very linked to the soil conditions and the microbial communities. And so that's what we decided to look at. We decided to look at the associated bacterial to the roots of our transplant and our control meadows. And indeed, if we look at the bacterial community structure, we can see that the structure of the bacteria associated to reference meadow in red is very different from the transplant. But also that the community structure associated to the roots of the transplant on the iron staple in orange is more similar to the reference meadow than coconut fiber mat in brown and base element in purple. And then we looked at what are the main bacterial orders that are responsible for those differences in bacterial community structures. And there were basically two main bacterial orders responsible for those dissimilarities: the bacterial order Desulfobacterales and Chromatiales. Why is it important? Well, first of all, you can see that they are both highly abundant in natural meadow, while only Desulfobacterales is also highly abundant in iron staples, while they are almost absent in both base element and coconut fiber mat. And why those two bacterial orders are important is because they are already known to be worldwide important for all seagrasses in two main processes: sulfide detoxification, which is highly toxic to seagrasses, but also nitrogen fixation. We looked also for the differences between two donor sources. And first thing, we looked at if there were differences before transplantation, in dark blue T0, 2 years after transplantation, light green donor meadow counting, light blue stone fragments, and with natural meadow. And as you can see, there is a difference in the community structure before transplantation and 2 years after transplantation. And we kind of have a trend towards reaching a similar bacterial community structure as it is present in natural meadow. But 2 years is not enough to reach that structure. For the bacterial orders responsible for those differences, again, Desulfobacterales and Chromatiales were the main ones, highly abundant in natural meadow, while Chromatiales was a bit more abundant, as you can see in orange, in donor meadow cuttings compared to storm fragments. And this leads us to another question. We saw that there are some differences in abundance of two main bacterial orders responsible in key processes like nitrogen fixation. And we already know from previous studies that nitrogen can be an element challenging to absorb for transplants. So we decided to look at the nitrogen content in the leaves of our transplant in natural meadow to check if we could have some differences that could be related to both the root system, but also the bacterial communities. And well, the results were quite surprising because there were no significant differences between any transplantation methods, which was super surprising because there were strong differences first in root morphological traits, with a very developed root system in iron staples almost present in the two other transplantation methods, but also difference in bacterial communities. And this leads us to this hypothesis, is that there are different resource use strategies between transplantation methods. Well, first of all, this method allows a direct contact with the sediment between the rhizome and the sediment. So this direct contact allowed to be in contact with some beneficial bacteria that can be present in the soil. Those bacteria, they can, by different processes, production of metabolites, they can stimulate root development. As the root system is developing, then it can absorb the pore water nutrients present in the soil. As the root system is not completely developed as it should be in natural meadow, the poor water nutrients is directly allocated to the root system growth. And therefore, you have like this, a positive feedback loop. However, the two other methods, the base element, the coconut fiber mat, they provide a physical barrier between rhizomes and the soil. Therefore, you have no sediment contact. So no contact with beneficial bacteria that could stimulate root growth. Therefore, there are no stimulation of the root system development. So the nitrogen acquisition then can only be absorbed by the leaves, and therefore you just have a conservative strategy with no investment in the root system compared to the iron staple. And another observation, super interesting between the North sources, if we look at what is happening 2 years after transplantation, you can see that the stone fragments have a significantly lower nitrogen content compared to both natural meadow and donor meadow cutting. And if you look at that result and the one of bacteria, which was also performed 2 years after transplantation, well, you have a higher content of chromatiales in the donor meadow, in the reference meadow, sorry, then the donor meadow cuttings, and then the stone fragment, which leads us to the hypothesis, but that will need further research to check this, is the higher abundance of chromatiales in the roots could lead to higher nitrogen content in the seagrass tissues. Conclusion of those 4 years of work and the two main objectives, they are the following. First one, do the stone fragments perform as well as the donor meadow cuttings? Well, morphologically, yes. However, if we look at the bacteria and the physiological and biochemical content, at least for nitrogen that I presented here, well, we can see that the donor meadow cuttings, they perform better than the stone fragments, but that this better performance does not seem to longer than the 2 years. So there seems to be a stabilization in performance over time. And then, which transplantation methods performs best? Well, we saw that the iron staple provided very good result if the hydrodynamic activity is not too high. But if you look at other ecological parameters like root system development, bacterial communities, and the cost, which is quite low also for this method, well, they perform very well. And then the base elements, they also provided very good survival rate even under higher hydrodynamic activity. But however, for different other ecological and economic indicators, they perform very poorly. So which leads us to emphasize the use of iron staple for seagrass transplantation, at least for Posidonia. If you want some more information of what I shortly presented here, you can find much more details in those 3 published presentations, and thank you for your attention.
Thank you very much.
Okay. Thanks, Arno. Let's go to move to the networking session and discussion. Then let's try to show the questions. Okay, that's here. Okay. Let's see. Let's try to answer the ones that are already here, and then we can do more questions for the in-person in the room. Then for Ariana, how is development measured as a response variable to measure restoration success? What exactly is it measured in the different studies? Ariana? Are you?
Yeah, I'm seeing.
Yes, sorry, I'm trying to share the screen if it's possible.
Okay, thank you, Ariana. Yeah.
Yes, for the plant traits, several explanatory variables measured for each survey for above and below ground tissues. For the development includes the growth data. So basically it's a ratio. Okay, so for the documents that collected and measure data on ratio on development, gross data were considered as development. Of course, for the above and below ground tissues.
Okay, thank you, Elena. This one for Luca. At what depth was your transplantation project?
The transplantation project, I mean, if you meet the transplantation initiative, was about 10-meter depth. The donor set a little bit less, and yeah, also the experiment around 10 to 13 meters, the two sites in this range, really close to each other.
Okay, thank you, Luca. For Fabio, there's a comment, very interesting that you work with seedlings. Are these areas you work in where seedlings are available every year?
Yeah, hello. Well, yeah, that was kind of the point. It's true that every year, of course, changing in quantity, but for example, in the case of Italy, in Sicily is almost a yearly production. You can see seeds arriving to the coast almost every year. That also happens not so much, but for example, in the Balearic Islands in Spain, it's much more common to find seeds than, for example, in the continental part. So that's kind of the point. There are areas which are more productive. Those areas might change from year to year, and to assess if this area, these highly productive areas, can be sourced for other areas in which sexual reproduction is less common.
Okay, thank you. Fabio, new one for you. How long and what is the technique to move the seed from one place to another and still be available to germinate?
Well, we collected seeds manually and in the different areas I saw in the map, and then we brought all of them to the lab in mesocons conditions. In fact, we had the seedlings germinating all in the same conditions, also in order to perform an acclimation to make sure that all seedlings arrive to the transplantation point in the same conditions and there are no like transportation related stress. We had them in this case, for 6 months in the mesocosm, and collection is just collected from the beach, not sampling, but from the beach cast seeds.
Well, thank you, Fabio. New one for Luca. Have you assessed different type of substrates for the survival, like different kind of mesh, without mesh, etc.?
No, actually, no. We actually rely on literature and iron staples, which is based according to the most recent literature, provided a nice survival showing the midterm, and also was quite easy to implement and to scale up in case for the restoration initiative.
Okay, two more for you, Luca. If pressure must be reduced before restoration, how do you deal with persistent pressures such as head waves or turbidity?
That's a really broad key question. You cannot deal. This is a really, recurrent topic in ecology, that is global versus local stressor. You can ideally manage global one, like heat waves that occur at a large spatial scale, rather than you can merge turbidity. It depends on the source of turbidity, of course, if it's to rainfall and sediments accumulation and sediment rainfall in the coastal area. But anyway, I would combine this experiment and also what we have done with looking for thermal refugia. So you can also combine identifying these recipient sites for restoration in combination with thermal refugia. You cannot deal with this with global stress, so like heat waves, but at least you can combine experimentation in the first tailored experimentation to see the suitability, along with analyzing framework mean to identify thermal refugia. I hope I answered the question.
Okay, thank you. Anyone Thanks. She has acknowledged your presentation. And the question is about how you measure the different parameters in order to determine how effective the restoration is.
I measure inside the shoot density, live shoot per patches, and the survival, the photosynthetic efficiency, probably with the using underwater fluorimeter, and the light adaptation, so not with the dark adaptation, so light condition, not dark adaptation. The surface area index, leaf index, and the leaf production using the puncture method, where we mark an hole 3 centimeters above the ligule, and then we measure the elongation the year after.
Okay. Now for Arnaud, is there a way to know how old are the storm fragments? It can be a factor to take into consideration.
Well, as far as I know, with a non-destructive method, there is no way to know the age of the fragments. You have lepidrochronology, but you need to remove the scale. So I don't really see how it can be done on, and also regarding restoration probability, because it won't just be possible to do that for all transplants. But yeah, it's definitely could be one of the major factors, the age of the fragment. This could have some issue. However, we had also an hypothesis regarding that at the beginning, is that the storm fragments, maybe they could perform less well than the cuttings, because they can be drifting for 1, 2, or 3 years, and they could be only surviving by using their carbohydrates, so the sugar storage in the rhizome, and they are just alive because they're consuming those reserves. But once they are completely consumes their internal reserve, they are not able to replenish it. So there was one of the original hypotheses, and so we did measurement of carbohydrate storage during the 3 years of transplantation. And what we found out is that they are able to replenish the carbohydrate storage as well as the nomadal cuttings. So I'm not so sure that the age would be one of the main effects regarding the performance of those fragments.
Okay, we have 2 more for you. There is this assistant called Anonymous, that is who is more asking questions. When you put the fragment on the cocoa mat, do you make holes where to put the cuttings? It is worth it look at the technique.
No, we didn't do holes. So the cuttings were placed on top of the coconut fiber mats, and they were tied with a zip ties. I know that in Italy, maybe it has very often be used the coconut mat, and on top of it a metallic wire grid, which probably increase the stability and reduce the detachment of the cuttings. So for sure, you can have higher survival rates with that technique. However, well, if you want to do holes in the coconut fiber mat, I think it can be quite labor intensive and bit complicated to put the cuttings, each cutting under the coconut mat, and by placing it always on top, I think you will often have a delay, at least in root system development, as we've seen with the base elements.
Okay, and the last one in the chat. Could be base elements and coconut fiber be used differently, allowing the plants to touch the soil? What was this not preferred?
Well, because logistically, it's feasible, but it's super labor intensive. Well, the base elements for Posidonia and how the design is currently made, it's not possible because the foliar shoots are too thick to fit within the element, and would be just too labor intensive to insert each foliar shoot of each cutting through the grid. And it's exactly the same with the coconut fiber mat. So I don't really see the assets of doing it, because it will be more labor intensive than just using the iron staple, which also are much more cheaper than base elements and coconut fiber mats.
Okay, thank you. Let's move on now for the questions here in person. We have some minutes before the coffee break. Yeah, please.
I have a couple of questions for different people.
Yeah.
So I would like to start with Ariana.
Ariana, yeah.
Ariana, in your guys' lit review, you mentioned replanting over dead mat as being seen as a not very positive, so as a negative, if I'm not wrong? Yeah, is that so?
Yes, actually, the meta-analysis revealed that planting on living meadow brought to unsuccessful outcomes. In fact, what I interpreting these results, I would say that because of a competition of a natural living meadow, of course. So it would be quite, I would say, very, very simple to say, do not transplant near living meadows. But I mean, there are some techniques which are nowadays considering transplanting inside little patches inside living meadows. So that's why I didn't mention that mud as an unsuccessful substrate. Of course, that mud could be a successful substrate.
Because it's more like not just a question, but more of a comment. I believe that when we look at restoration, we are not here to play God and substitute a different type of substrate should be there. Every type of ecosystem and substrate has its functionality when we look at the environmental patchiness. So why will be, like, if there was a mat there previously, if there was a seagrass meadow previously, it means it is naturally successful. Mm-hmm. And in restoration, we're here to help it survive, not just choose where we want. So looking at then choosing other types of substrates, we're just then choosing how we want our underwater scenery to look like. But that there is a sandy patch doesn't mean it's useful. Actually, sandy patches are super necessary, and we can see with Timothea nodosa that it's actually starting to stretch out, and it helps stabilize substrate, which later allows Posidonia oceanica to grow over it. So, for example, we always try to replant over mat that is near living prairies, and we actually have high successful rates of survival. So I just would be really careful on saying, like, to consider not planting on mat, because then we're just choosing to alter our underwater seascape to our liking and not respecting what has naturally occurred there. And then, Craig, I have questions for you as well. Really cool, really interesting approach. What are your pods planted in? So you guys have the structure, and then you put the plants in.
Oh, but the proof of concept, it was simply build a sand, just to see.
Sand. And then the idea is once they develop the roots, then you would put it in the soil.
That is a good question. Yes. How that's done will depend on a few different things. So we don't have a final system for once we've got a nice big pod full of stuff, we don't have the next sort of link for how to do it. Now we have a few ideas. This was not just generated for Posidonia, which is side ground, we're thinking more along the lines of Sunbio and Zostra. In that case, we have a couple of options. You could either have big trays that could be lifted out, put into place. Maybe the whole thing could be slid off. Maybe you could just use it as a source of bugs or plants so that you don't have to go to natural meadows. It's like your donor source. So that's kind of the idea, is that once we get to the stage where we know we can reliably grow different places, different species, okay, how are we going to use this? Best use this. We see it as a tool for doing restoration, minimizing impact on donor meadows, just trying to work out what works best.
It's really interesting because like you guys are obviously taking out of the equation all like stressful factors like sedimentation, physical impact over like when those cuttings or seedlings try to grow. But then when we introduce it into the natural medium, those stressful factors will play.
But that's one of the things that we were planning to do with the idea of having a smart pod. So with the meta-analysis, for example, the deeper to shallower transplants do better. Well, if we can drop the pod down to stress it, or if you want to put up to heat to stress it, this is, you can use it as an experiment. If you've got 10 pods, put 5 up to heat stress them, or light stress and see which one works, develop a strategy. It's an ongoing iterative process in my mind, but I'm an experimentalist.
Thank you. And I got a last question.
Yeah,
Can I
sure.
take advantage of the role?
Yeah.
Arno?
Yes.
Really interesting project that you guys did and super interesting insight on the bacterial community, which I think is something that we generally tend to forget to look at. And it was also really interesting that you guys looked at the comparison between cutting a healthy meadow and broken fragments. Nonetheless, my concerns come again. Why, if we're trying to restore and help an ecosystem recover, why are we trying to cut it and diminish, make it go back, no? So really cool that you guys found that there's actually no significant differences and that restoration experiments don't try to target cutting more meadow to try and replant it without actually guaranteeing the success of its growth. So I'm pretty happy that you guys found that there's no successful differences, significant differences there.
Sorry, could you repeat? I have some noise I didn't hear.
No worries. Basically, it's again concerned because at the start of looking at Posidonia restoration, a lot of the experiments and programs looked at actually cutting out sods of perfectly healthy prairies and then replanting it somewhere that was decided. And then your guys' experiment looking at cuttings of a perfectly healthy meadow to then try and replant it somewhere where you actually don't know if it will be successful, which great, it's an experiment, and I'm really happy that you guys found that there was no significant differences to not actually encourage people to start cutting our meadows that actually need to keep on growing and not being cut back. But I definitely would say it is really interesting for all these projects. As you guys know, here in the Balearic Islands, we have got the electrical cable being brought into Algudia, Pudenza, and when looking at how to do it, we have to cut through seagrass meadows. So maybe a really cool experiment I propose here is to look at where that cable has to come through and they have to cut out the sods, look at areas to actually replant those sods fully and not just destroy, but kind of like what you guys proposed. So yeah, thank you very much for all these presentations.
Thank you.
Thank you.
Have questions?
I already typed it into the
Okay, great. You was anonymous.
Yeah.
Okay, let's move now for the coffee break. Okay.
So see you for the online participants. See you on 11:30.
11:30 is now.
Okay, welcome back again to the second part of the workshop. Let's move on now with the next presentation that will be for Frederick. Let me introduce Frederick. Frederick holds a Bachelor in Agricultural Engineering at UPC and a Master Science in Applied Mathematics in Technical University of Delft, specializing in numerical modeling. Now, Frederick at UPC is focused on developing low-cost methods for replanting Posidonia oceanica.
How do I click to the next?
Joaquin is the controller.
Okay. Hello. So, as Joaquin mentioned previously, we at SARTI are more a group of technologists than biologists. So why do we care about Posidonia? Shall I continue or shall I wait?
Yes, please.
Yeah. The reason we're interested in Posidonia is because we've got an underwater observatory at 20 meters depth here, 4 kilometers out to sea. We've got a laboratory here, and the two are connected by a cable which runs across a Posidonia network, the network meadow, and the meadow has about 700 square meters. The meadow is 700 hectares big, so it's pretty big. So here, can I go back? Can I go back?
Yeah.
How do I go back?
With the arrows, yeah.
Here we go. Here.
If you want, I can change for you, Frederick.
No, it's okay. It's okay. I got it now. Okay, so in 2022, we started with our first Posidonia projects. And the first Posidonia project was a cartographic study of the degradation that had occurred in the meadow. So we only had one map at the time. We compared it with the data. The map is from 2011. And we compared it to data from the fishermen to see how much degradation there had been. And on the Earth side, it was not so bad. On the sea side, there was quite a bit. So the yellow dots are where the fishermen say the end of Posidonia is. There's quite about 100 meters, which had been wiped away by storms and stuff. While we were doing the study, we were notified that a massive flowering was going to occur in Ametje de Mar, 100 kilometers down the coast. So for the fun of it, we decided, why don't we go down to the beach, pick up some seeds, we ask for permission from the Generalitat, and then we stick them in fish tanks to see what happens. The first question was, are we able to germinate these seeds? And the answer is yes. We stuck them in fish tanks. We stuck them on carbonate rocks to see what would happen, because the literature, thank you Arianna Passini and all her colleagues and the studies that have been done, said that fixed substrate was a good place to propagate because the roots would stick. In this way, we were able to grow out the seedlings onto the rocks. Some of them let go, some of them attached quite nicely. But then in the course of 7 months, we had vigorous growth to about 10 or 15 centimeters of leaves. And we were able to verify that the roots are sticky. So they stick to pebbles, they stick to rocks. And then if you have little plants stuck to rocks, you've got a little block of replantable material which you can throw into the sea without having to dive. That was kind of the idea of the first pilot projects. So this is what they look like when they're falling. The leaves act as they keep them with the roots face down. This is a pretty picture. So this was after the rocks had all settled. We gathered them together and we put them in one safe place. And then we watched to see what would happen over time. The first experiment didn't go very well. We had a couple of survivors, but lots of the rocks tumbled over with the storms. So we have one survivor standing up. We actually had one block that tumbled over, and the plant still continued to grow, even though it was face down, which was kind of funny. But let me see. So yeah. Okay, I got it already. So we were left with two questions. Number one, we need to improve the mechanism for throwing rocks so that they land and stay at the bottom properly, basically make them bigger and more solid. And that's the work that's going on, being done by Alex, and he's going to present that in the next presentation. The second thing we need to figure out is we need to have a regular supply of seeds, because just the year after that we used rhizomes, and they don't grow as vigorously. So I thought, okay, well, if we are able to produce seeds ex situ in tanks, then that might be interesting. So those are the two experiments we're running at the UPC at the minute. Okay. Is it possible to induce flowering ex situ in plants? One example of a trial done in our colleagues in the Instituto Español de Oceanografía, Oceanographic Institute of Spain, said yes, they were able to heat stress plants that were in tanks. They applied a heat stress of 4 degrees, and they got flowering reaction. They didn't continue on to the actual seed production stage, so it's kind of a green field for us. And then there's also lots of, there's a couple of papers, including by Patricia Schitz and her colleagues. She's also going to be speaking later. That what happens is, if in the wild environment, if you see a heat shock, it produces a flowering event. Right. So with that little pilot results, we were able to go to the fisheries and ask for a grant to set up a little laboratory. So we have a little laboratory with 5 tanks. It's covered, and that's a place where we can do our activities. Yeah, so in this one, the first one, we've installed a series of sensors to monitor the temperature, conductivity, the pH, and the oxygen in one of the tanks. It's also proved important to install an automatic system to apply distilled water when necessary. Here you see a diagram version of our setup of tanks, sensors, and temperature regulation. And then for the experiment, we're going to use one as a control and the other one as the test case. This setup, we ran a trial last year just to see how it would go with the sensors. And one of the tanks had, as a test, we controlled the temperature. So as the summer came in, we lowered the temperature to 35 degrees Celsius. No, that can't be right. Okay. We set it to not too hot. Here we go, this is temperature. Temperature is controlled, and that leads to a less mortality rate. Whereas if you let the temperature go free, you have a 100% mortality rate and all of your plants die. Then we repopulated the tank just to see what would happen with controlled temperature, and we had a more reasonable survival rate. So what are we going to do with this setup? One or two of the tanks we're going to do a heat stress, and one or two tanks we're going to apply control environments. So this is the temperature. The red line is the temperature regime that we force on the tanks. And then in August or September, during 6 weeks, we're going to apply a temperature shock of 4 degrees. And then we're going to sit back and hope that flowers grow. Thank you very much.
Questions or later?
I think better late. You can use the anonymous for asking.
No.
Okay.
You're not going to do the you know,
Okay, then let's move on. The next presentation will be for Alex. Alex holds a degree in marine science and technologies from UPC. He is now pursuing a master's degree on oceanography and marine environmental management, specializing in marine biology at the University of Barcelona. His final degree project focused on the developing an innovative low-cost method for replanting Posidonia oceanica, and then he has been working at the SARTI laboratory, monitoring the success of these restoration techniques and developing new scalable methodologies for marine regeneration. Okay, Alex, the floor is yours.
Thank you. Hello, everyone. My name is Alex, and well, as Joaquin said, I've been working here for almost a year, working with Frederick in searching some methodologies for developing the restoration of Posidonia oceanica, and this I'm going to talk about. We're going to present you the Posibox, a low-cost innovation methodology for Posidonia oceanica restoration. First of all, I want to present you our Posidonia medal here in Vilanova, as Frederick said before. This is our medal, and here at the end of this line we have the UPC Undercable Observatory. So the main characteristics of this medal is that it has 516 hectares of extension, and the depth goes from 10 to 16 meters. So the existence of UPC here in this Posidonia medal is very useful for us in order to control our experimental fields and have and we can monitor these experimental fields thanks to the UPC. As you can see here, we've developed two experimental fields. Here is the one that is currently working now, and this is the one that we used last year. So before presenting the Posidocs, I want to talk about the restoration nowadays. So from our point of view, restoration nowadays have two things in common. The first one is that in plenty all of the restoration methods, the use of divers is included. So this use of divers to do the traditional restorations involves a high cost in these techniques. So here I collected 3 different papers where we can see restorations that go from 100,000 euros to maybe almost 1 million. So from SARTI, we wanted to develop a low-cost strategy and a methodology that eliminates the use of divers in order to do a restoration. So for that reason, we created the Posidocs. The Posidocs, as I said, are created in order to eliminate the use of divers. So it's a simple structure that it's made, the base is made by a calcarean rock. So we attach to this calcarean rock 4 Posidonia plants collected from uprooted even storms and collected from the beach. And then we attach these Posidonia plants with cotton or hemp ropes. So now I'm going to explain a little bit the methodology that we use to collect and to do the deployment of this methodology. So all the Posidonia that we used for this experiment was collected from Ametlla de Mar, a town in the south of Catalonia. Why Ametlla de Mar? Because there they have the biggest Posidonia meadow in the Catalan coast, and also the characteristics are very good in order to collect Posidonia plants from beaches, because the Posidonia meadow is less deeper than here we have here in Vilanova, and also is closer to shore. So the storms and the waves had a bigger impact there, and it's easy to find Posidonia plants after the storm events. So we collected them there in Ametlla de Mar, and then we keep them in aquariums of salt water, as Frederick explained before. So after this, we create the Posidocs, and we go to the deployment phase. So here you can see some video of how we made the deployment, some pictures. So as we the objective is to reduce costs and emissions. So we throw the Posidocs from the surface, and they land to the seabed. Here you can see more videos as they fall into the water column. As you can see here, the characteristics of the Posidocs were very successful in order that they fall very vertical and very stable, most of them. So, and then here we can see how they fall and they stay in the seabed. So now we are going to talk about the first results that we obtained. So first of all, say that this is a pilot study, a first experimental stage. So we don't have a very big sample, a very big amount of data, but with the result we had, we can say, well, we throw 10 Posidocs, and the 40 Posidonia plants that were attached to the Posidocs after 7 months of the deployment, they were still there. So the materials that we used were very successful. And the most important statistic that we can see here, this is survival rate. So after 7 months in sea, we observed a 55% survival rate. That is pretty good for the first 7 months. And as you can see here, the statistic for each Posidoc, we can see that it has a high viability. And then also we used the amount of leaf of each Posidonia plant in order to determine their state of health. So after 7 months, we've seen that Posidonia plants that had more than 7 leaves in the rhizome had a high probability to survive into the sea. So this statistic is very important for us in order to be more precise at the moment of recollecting the Posidonia plants in the beaches uprooted by the storms. So finally, we are going to talk about the economic impact. So it was one of our objectives in this methodology. And as you can see here, it's a simple table about the cost of the production of one Posi rock. And as you can see here, the cost is less than 2 euros. It's actually 1.96. And with that data, we make an approximation and a comparison with another experiment that, well, Get's experiment that it was an experiment that tried to do a low-cost replantation as well, but using a traditional typing methodology. So as you can see here, the 50% of the total cost of the experiment was used for the transplanting cost, that it was mostly dedicated to the divers, and also a big part of the positioning cost. So if we reduce these two costs, we arrive to a reduction of the 70% of the total cost. So with that information and with our calculations of our cost in the production of these Posi rocks, we've seen that we can replicate a similar restoration, trying to achieve the same area, 1,000 square meters of replantation, with a budget of 21,000 euros, more or less. So to end with this presentation, I want to highlight the thing that I said at the beginning of the result, that this is a very first stage experiment, a pilot study. So we are now trying to do an experiment larger and with more scale. But with the result that we have, we can talk about biological success, because we obtained more than a 50% of survival rate, and also we detected a proxy in order to pick the better Posidonia plants when they arrive to the beaches. Also, we can see that theoretically we calculated that it is possible to reduce the cost, but we are very strongly positive in this reduction of costs in order to eliminate divers. And also, we are using natural materials. The Posi rocks are natural material. And also, we are collecting Posidonia plants from uprooted meadows after events. So we are promoting the circular economy. And to end, I wanted to show you the next experiments that we are going to develop into the UPC meadow. And as I said, we have already prepared a bigger scale of Posi rocks in order to improve our results and improve our data and information. And also, we prepare a camera in order to monitor this experiment in real time. And also, we are doing some traditional restoration to compare the results with the result that we are going to obtain with these Posi rocks. So thank you for your attention.
Thank you, Alex. We get again on time. Thank you. Yeah. Now, next presenter is Albert. Alex. Let's move to the next presentation. Albert received his bachelor's degree in marine science and technologies from UPC and a master's degree in advanced physics and applied mathematics with a specialization in geophysical fluid dynamics from the University of Balearic Islands. During his master's studies, he completed an internship on the beach monitoring department at the observing forecasting system that is called SOFIP. And he is now a PhD student at UPC, where he is trying to apply studying AI methods for Posidonia monitoring. The research interests include new technological advances in marine and coastal monitoring, ranging from computer vision to marine robotics.
Joaquin, maybe better to end the acrobat and the task.
Yes, like,
Not only the whole. The arrows, it's the arrows, just the arrows.
okay, thank you. Okay, well,
Okay. Well,
But we're not saying.
bueno, sharing.
okay. Well, as Joaquin said, I'm Albert Catalan. I'm a PhD student at UPC, and I will talk about using satellite image and AI for sea-grass monitoring. So, what I'll be talking will be more about where to do the restoration, and the plan is to use satellite images to get an historical data about meadows to see which parts of the meadow are degrading and which are going better, in order to put the restoration in the parts that are going better and not the parts that are losing Posidonia. This is a little bit what I'll be talking about. And we'll start for the study case. I'll also talk about the Garraf meadow as my colleagues Frederic and Alex. And here we have 3 meadows, which everyone is a bit different. For example, the Cubellas meadows is a pretty dense meadow, and it's low depth, so it's really good for satellite image. This one, it's the Sitges meadow, and it has a really low density, so it's pretty tricky to pick up with satellite. And the Vilanova meadow, it ranges until 17 meters of depth, making it really difficult to detect, especially the lower limit, and it fuses a little bit with the deep water. So you have to do a lot of image processing to get that lower limit correct.
And these are like the challenges of this site, and one of the reasons that I chose it. So now we'll talk about a little bit of the workflow and how we do this to go from an image from Sentinel to historic evaluation of the middle. So we start with images from Sentinel, the Sentinel satellite. Then we do image processing to better see the Posidonia middle in Sentinel. And then we pass an AI detection model, in this case, SAM 2.1. We do a little bit of quality control for the detection, as they were done by an AI and not by hand. And then with that, we can do a temporal analysis. All these steps will be better explained right now. So for example, for the satellite, the one we chose is Sentinel-2 L2A. So this gives us a bottom atmosphere surface reflections, which means it's corrected for atmosphere, meaning that all images should be equal despite the atmospheric conditions. This is really important, as if you don't have this into account, images change for things that are not related to the middle. Moreover, this satellite gives us images from 2015 to today. So you have an historic of 10 years, which could be better, but you have a 10 meter pixel resolution, making it really, really good. And a part of this, it's not an RGB image. You have multibands, letting you do a lot of post-processing way better if you only have red, green, and blue. And well, it's also an open source satellite, meaning everyone can use it. It has a lot of coverage, and it comes every more or less 5 days, giving you a lot of data. And in that way, you can not use images. As you have so many images and Posidonia growth is slow, you can pick only the best images. For example, in our case, all images with more than 10% clouds in the images were instantaneously discarded, as they would give bad results and are not that useful. So, as I said, I'll try not to enter into a lot of specifics, as it's a lot of mathematics and there's no need. There's a lot of image processing, as you can see really right any Posidonia middle here. So what we do is we take these 4 bands of Sentinel, the blue, the green, the red, and the near infrared. And the first thing we do is, if something has more than 0.1 near infrared, we paint it white. This helps us. This means we paint white all land and any cloud. Land is not so important, as we are not detecting land, but it gives less complicated structures to the AI model. And the clouds, white is really easy to detect for AI as something completely different from anything near. So that's one step. The next step is red. We put it at 0, as Posidonia does not have red. And then we do a little bit of processing. I won't enter much why we do this, but it's like for green, as Posidonia absorbs red for photosynthesis, we do a bit of processing to better show Posidonia. And this is how an image looks before and after processing. With this light, it's not really easy to see, but here there should be a Posidonia that you can see really clear. Well, here it's a bit better. So you have here for the main middle, the Vilanova middle, and here, as you can see, it's way easier to see. You can even see this one and this one, while in this one, it's way worse. So that's a good first step. However, it's not good enough. So we do more image processing. In this case, we do a Clahe image processing. which stands for contrast limited adaptive histogram equalization. It's the same thing out of mathematics. The basic is we have this histogram of pixels ranging from 0 to 200. Well, the first thing is we pass from RGB color, red, green, blue, to lamp, which separates luminescence from color. And we only touch luminescence. As our main problem is it's in deep waters. Most studies are done around 5 meters, and we're studying middles that are 10 meters deep and even 17. So we have a lot of trouble with the merging of deep water and Posidonia, as both are really dark. So we only touch luminescence. And what we do is these histograms of luminescence that go from 0 to 75 with 8 matrices, we expand them to go from 0 to 150, making it way clearer. It would be similar to a Gauss campaign that you can do on scores of students or something similar, but for luminescence. Making this image, that same thing here, it's better. You can barely see the Posidonia middle. Now it's clearly seen, and it's way better with the lower limit, where it has a little bit of white beneath, and this helps a lot our segmentation models. As for the AI, we use SUM 2.1. As I said, this is a model from Facebook, and being from Facebook, that's a big company, it has a lot of data. So the good thing about this model is you don't have to train your images. It's already trained, so you only give the images points where the middle should be, and it gives you detection without having to do training. Well, it's a state-of-the-art model and all of that. And yeah, in our case, here we have an example. What we give it. We give it a bonding, we do separate detection for every middle. This is important for the post-processing that we'll do later. But you give it a bounding box of where the middle is going to be and where is it going to do the detection. Then you give it positive points, points where there's middle, negative points, points where there's no middle, and then I draw a silhouette around the middle with a little bit of margin that's going to be useful. This is not for SUM, the silhouette is for doing quality control of the detections. And for example, in this case, there's only one positive point. I did this because I know this is going to disappear later. So if I put here a point saying here's middle, maybe here's middle in 2015, but in 2020, this middle disappears and the model starts having trouble because I said that there's middle where there really isn't. Then about the quality control, using the silhouettes I've showed you around the middle, what we do is basically, if a 10% of the detection is bigger than the area of the middle, the detection counts as invalid, but only for that middle. For example, here it's a clear case of bad detections because there's a lot of noise in the form of clouds. However, it could happen that there's a lot of clouds here and you can clearly see this middle. So, by separating into 3 different detections, you can have no data from here and data from here, or on another day, no data from here and data from here, which is pretty useful as more data is always better. And moreover, what we can do is if the detection is deemed as valid, all the data, all the detection pixels that are outside of that silhouette are also discarded. Because, for example, there are some cases, you can see it here, but for example, a boat goes through this middle. And then it takes the boat's passage as part of the middle. So you do some post-processing and say, this is not middle. And well, after that, with you have a lot of masks of where's Posidonia during 10 years, so you can do some analysis. The first thing we did is a temporal comparison, where we can see all the meadows are pretty stable during all years, which is to be expected as these are pretty dense meadows, and a lot of loss is prior to 2015. However, there was one meadow that has a lot of fall-off. It's this one, which would be this one. This is an image that compares the first 2 years, meaning 2015 and 2016. I know all the history with the last 2 years. The first years with the last 2 years, first 2 years with the last 2 years. And this is, we use 2 years as only 1 year. You can have not so good detections. And by using 2 years, we only say that a place has Posidonia in all the detections if it's in more than 30% of the images on the year. And here we can see there's a clear loss in this middle. And the more interesting thing that we can see is in this middle that's really stable, there's a clear regression in the side that's nearer the coast. However, there's a little bit of extension where you're farther apart from the coast, which is a little bit counterintuitive, in my opinion, as I would assume that the part that first touches the waves would be the one that degrades the most, but this is to be proved that that's not. This is similar to this. This uses all data from all over detection and shows the percentage of times this pixel shows with Posidonia. And here you can see this really clear loss. And here we have 2 problems. We don't have this is shown as it's as if it's lost. However, the problem with this is these are really low density Posidonia meadows, this part and all this middle. So what really happens is not that we lost this part, which also happened, but the main problem of this is in really, really good days, it detects it. And in days that are really good, but not as good, it detects everything except this part, which is one of the main problems that this approach has. Another really good thing this has is when you have stuff like, for example, the generated bathymetries, which are really high quality, but you have a date from 2011 and a date from 2024 with a lot of quality, but a lot of time has passed between. But here you can compare years between each year. So for example, here we can compare the year 2019 with the year 2020. And here is where this huge loss in the Cubellas meadow happened. This also is when the Gloria extreme weather event happened here in Catalonia, meaning that it's really possible that this loss is due to Gloria. And well, some conclusions is that this is a pretty reliable tool for monitoring. It has the good part that you can look at extreme weather events because you have more data than with other sources, not so high quality, but a lot more data. You can make an historic of 10 years, which is really good right now, and will only get better as you will always have this data, and satellites will only get better. And well, it's even better for lower depth Posidonia. Like this is a pretty hard case for this satellite detection method. As I said, most of the studies were done between 5 and 10 meters, and this is at 17, which is a real challenge. However, as everything, it has its limitations. The main thing I said is till 2015 is good, but it's not the best. And it really struggles with low density zones and also the boundary areas, which are the most important, is also where SAM has most trouble. So it's like you have these detections, but are you 100% sure that you can trust these detections, which happens always with AI? And well, that would be all. And okay. Thank you for that.
Okay,
Thanks very much.
okay. Now let's move on to a new presentation that will be, in this case, online.
Hi, everyone.
Hi, Patricia.
Hi, Patricia.
Good morning, everyone. Thank you very much for organizing this workshop. So interesting so far. So today I'm gonna talk a little bit about how some experiments can provide information about seagrass restoration. And in particular, I want to talk about an experiment that I have conducted in 2023 with seedlings. So as everyone know, and also Frederick, thank you very much for citing this article before, the flowering of Posidonia oceanica is increasing, and recently it has been associated with the increase of sea surface temperature. So according to a correlative experiments, we found out that the sea surface temperature in summertime not only was increasing the inflorescence density, but also the probability of living fruits. In another experiment, in another correlative study, we were also wondering if only the summer temperature is the one who is affecting the flowering. Because if you think about the process of the flower induction, it actually started in autumn, the year before we see the flowering. So again, through a correlative approach conducted in different study sites in Sardinia, we were investigating not only the temperature of summertime that again confirm the same results as before. So the highest is the temperature, the highest is the number of flowers and therefore fruits and seeds, but also the temperature that the meadows experience in autumn time the year before. So if you see at the graph, the sea surface temperature range, the more it increases, the more we have the probability of flowering in the summer after, and also the longest marine heat wave we have in autumn time, the highest is the probability we have more flower in summertime. This is because autumn is the actual moment in which the plant is starting the whole process of making the flowers. So it's not only important the sea surface temperature in summertime, but also in autumn. And so because the temperature is increasing not only in summertime, but actually all year along, we are assisting to an increase of flowering events, and therefore also lots of seeds and fruits that are beached on the sea. So in 2023, as mentioned already before, there was a massive flowering events all along the Mediterranean Sea. And the situation on the beach were these ones, or even more. There are some pictures where you can see like so lots of fruits and seeds all along the beach. And actually, a new paper about the massive flowering events in 2023 I just released last week from Patrick Astruc. So we take the advantage of collect some fruits and seeds to provide information about the restoration. So the goal of the study was to provide information for future restoration action using the seedlings, since this material is increasing and will increase also more in the future. Going to assess if the different thermal environments affect the growth of the seedlings themselves, but also if the density of the meadow can affect the growth of the seedlings themselves. So we have chosen two different coasts of Sardinia, the west coast and the east coast. The west coast is much colder than the east coast. And the main difference of temperature, you can see it in summertime. Also, the west coast of Sardinia is very famous for the Mistral wind, which is a wind that is coming and normally lasts for 3 days and is quite strong. So it goes to remix all the water from the surface to deepest. So it's always colder when a Mistral wind comes. So between April and May 2023, we were able to collect more than 500 seedlings. We left them germinate in the water, And when we started the germination time, which was June the 10th, we had a total of 416 healthy seedlings. There were collected 240 from the West Coast and 176 collected on the East Coast. So for each group, half were left germinated on the origin coast, and the other half were left germinating on the other coast, so conducted a cross-site transplantation. The seeds were left germinated 3 meters deep, as you can see in some nets, and just in a natural environment. When it was the time of the experiment, so after 1 month and a half of germination, we left in each side half seedlings from the origin and half from the other side, but also we have divided them in different density meadow. So we put some of them on the dead mat, so where they could have a very high lighting radiance and where the hydrodynamic forces are much higher because there is no protection of the meadow itself. We put them at 25 and 50% of density, and we actually created this density just by cutting the leaf bundle for the 25% of 3 shoots out of 4, and for the 50% of density in 1 shoot out of 2. And then we also put the seedlings in a untouched meadow, so density 100%. As you can see from the picture, the seedlings were not put directly on the substrate, but they were put in some plastic pot surrounded by some stones where they could actually attach the roots, and they were covered in a way to avoid the loss of material. Also, the pot were covered by herbivory nets, and as you can see from the picture, there is also like a smaller nets around to avoid any herbivores pressure. The variables that we consider were the morphology in terms of total number of leaves, total leaf length, maximum root length and the number of roots, and also the total length of the necrotic brown tissue. Also, we evaluated the mortality and the leaf fiber content. Leaf fiber content is often used as a proxy of resistance to herbivores, but also resistance to the hydrodynamic forces. Here there are some pictures of the plastic pots put inside the meadow, so you can see that it's actually like very hard environmental conditions, no lights, but also a little bit more protective from herbivores. For the whole experimental period, we put some temperature logger. So the difference in temperature was statistically significant. As you can see in the East Coast, apart from the first part of the summertime, was warmer for the whole time. And on the West Coast, you can actually see when the mistral wind that I was talking before was acting, because you can see this very low peak of temperature where the water is mixed from the surface to the deep. The graphics below show the light intensity in the cold and in the warm side, but you can see that the values are the same, and there is a very big difference as expected between 0, 25%, 50% to 100% of density. So in the first case, we were actually evaluating the acclimation of the seedlings, comparing the origin side and the development side. So what did we expect? Like if the thermal conditions are actually having more effect, seedlings, regardless of the origin, will grow at the same time according to the environmental conditions. If opposite, they will grow, seedlings from the same origin, they will grow in the same way, regardless to the development side. And what we found was a very similar results, actually the same results that Fabio found with these seedlings from Sicily and Campania that he was mentioning before. So let's say that the thermal conditions, the environmental conditions of the development side are not as much as important as likely a genetic component. So you can see that seedlings coming from the East Coast, so from the warm coast, they were actually having a higher number of leaves, higher total number of leaves, higher number of roots and maximum root length, regardless of where they were put for the time of the experiment. But also, again, what Fabio found, it was also something that we found, growth and mortality are having completely opposite pattern. So also in this way, the seedlings that were coming from the East Coast, that were the one who were growing the most, were also the one who show highest mortality. When we compared the fiber content, we found out that seedlings coming from the West Coast, or the cold coast, were the one that had a fiber content higher than the other seedlings. And then we justified because, as we mentioned before, the cost on the West Coast is affected by the mistral all the time. So it's kind of there is more circulation of the water, there is more hydrodynamic forces, and the seedling somehow has to have a higher fiber content to contest the hydrodynamic forces. When we were checking how the density of the meadows can affect the growth of the seedlings, we see something that we completely expected. So the maximum root length and the highest number of leaves were found at zero density or low density. Because at the end, this is the place, it's true that they are not really protected by the meadow themselves, but it's the moment where they have to, let's say, use their own energy to protect themselves. So they will have the need to produce longest roots to be attached to the substrate, and also the number of leaves, maybe they have more way to get the light radiance and produce more photosynthesis. In terms of mortality, we found a higher mortality inside the meadows, and for the fiber content, we found the highest fiber content in 25, 50, and 100 density. So inside the meadows, regardless the density compared to the dead mud. So in conclusion, the results today suggest that the thermal conditions of the receiving sites do not really affect the seedlings development during the first months, but there is likely a very important genetic component that might affect the growth of the seedlings. So again, I also support the local support theory that we mentioned before. However, if you want to have a higher number of seedlings, but with a lower growth, or if you want to have more developed, less developed seedlings, but with a high mortality, then it totally depends on the objective of the restoration. So this was for Sardinia restoration can provide information, but again can be different for every single local place. So it really depends on the objective of the restoration if you want to have more developed but less number of seedlings or not. However, what is certain is that to provide more functional seedlings to use in a restoration, seeds need to germinate at very low canopy density or in dead mud. Thank you very much.
That's Patrick.
Yeah, thank you, Patricia. Well, let's go to the next presentation. Let me remind you the Slido where you can put your questions for the next networking session. Now, the next presentation is for Fabio Badalamenti. Fabio is a marine ecologist at CNR, IARS in Palermo, an honorary professor at the University of Edinburgh. His research focuses on coastal marine ecosystems, biodiversity, and restoration, with particular attention to Posidonia oceanica Camilo. He has worked on seedling recruitment, plant-substrate interactions, root adhesion, and substrate-based approaches to seagrass restoration. His current interests include substrate-mediated recolonization and nature-based restoration of degraded seagrass habitats. Fabio, the floor is yours. Thank you.
Thank you very much for this introduction. I hope you can hear me pretty well. I changed a little bit the title of my presentation in Retention, Anchorage, and Establishment across Colonization and Restoration Stages in Posidonia oceanica. This is because I think retention, anchorage, and establishment are three important phases when we deal with Posidonia restoration. When, for example, we have project or we are looking at passive recovery, assisted recovery of active restoration, propagule retention is the first bottleneck we found. Without propagule retention, there is any restoration possible. So I would like to define retention as the temporary physical trapping of propagules by substrate features or structures. And I mean with that the roughness of substrates, the presence in the substrates of crevices, holes, boulders, stones, or vegetation. And trapping of propagules should be long enough for biological anchorage to begin. If a propagule, vegetative or sexual, is found, for example, on unconsolidated substrates such as sand, they will be drifting along the sea bottom, carried out by currents and waves, without little or no chances to get retained. If we consider instead consolidated substrates, there are plenty of caves, holes, and crevices of different size and dimension. So if we go underwater, and for example here, we see a vertical cliff underwater in one of the volcanic islands in Sicily. As you can see, is plenty of crevices and holes. And in the right side is the image of the same cliff a few hundred meters ahead, where possibly a seed had the chance of being retained but by one of the crevices, and a little patch of Posidonia is growing even on a vertical wall, even on a hard substrate with low sand in the surrounding. Another consolidated substrate which is interesting to work with are boulder fields generated by coastal cliff collapse. Here you have boulders of different size, and when they are joined one to another, they provide a huge amount of holes and crevices of different size. And this is a perfect place for a vegetative rhizome to get entangled and start a colonization, as we see on the right side of this slide. Boulders are interesting, and here I report the case of a trench dragged in Capofetto, Western Sicily. This trench was backfilled with stones, and again, not we, but the seagrass had the chance to colonize this area made by rubble mounds. So rhizome detached from the adjacent seagrass had the chance to enter inside the trench, and in a matter of few months got entangled and started to grow. After 7 years, more than 3 hectares of seagrass meadow recovered inside the trench, and where the Mount Valley plenty with these stones was present, more than 40% of the area was covered by Posidonia. And this is what happened after 10 or 15 years. I don't remember when these images were taken. The area of the rubble mound was not easy to be identified underwater, and as you can see in these red arrows, a little mat started to grow. Something similar has been reported in Ischia Island, where another trench was dredged. In this case, after 10 years, 12% of the surface of the trench was colonized by Posidonia oceanica. And of course, we are talking of a spontaneous colonization. Same things happen to seeds. When a seed sank on the consolidated substrates, he had the chance to be retained and grow a seedling. And when with consolidated substrates, I mean a rock or boulders or pebbles. These graphs show you a comparison of seeds sunk on consolidated and on unconsolidated substrates. The one sunk on consolidated substrates remained in place for months or years and formed seedlings. The one sunk on sand disappeared in a matter of a few days or a week. So after retention, and when retention is successful, we have a second important phase, which is anchorage. Anchorage is the development of physical or biological connection with the substrates through, for example, root, or we will see root hairs, rhizome, etc. As some in an earlier presentation, Boulanger, Arnaud, showed as one of the rare case of study of rooting after transplantation. I found very difficult to study rooting after transplanting a vegetative propagule. They can deepen the substrate, of course, they can envelope rocks, they can attach directly to rocks, but I prefer to study anchoring of roots on seedlings. In this case, we are talking of root adhesion. So what we have done, we have chosen two kind of blocks, one made of travertine and the other of calcarenitic stones. These are very frequent stones in Sicily and are stones up on where Posidonia grow. These blocks were 10 per 10 centimeters in size, and there were crevices along their length of 5 millimeters in width. What we observed after 5 months of cultivating these seeds on top of the blocks was the development of roots, and roots were following, as you can see here, the crevice. And roots were also able to start the enveloping the block completely. What is interesting to note is that roots were surrounded by this yellow slimy substance. And we can see better in this slide. Here we can see the root length, and the root is surrounded by this yellow thing. If we magnify this image, we discovered that this yellow slime is made by tens of thousands of root hair protruding from the root. The peculiarity of root hairs in seedlings is that once the tip of the root approach and get in contact with the substrates, they modify their shape and produce what we call a pad. And a pad has a lot of digitations, and the shape of the pad changes accordingly to the roughness of the substrate. If we look at the interface between the root hair pad and the substrates, we notice a substance we call a glue-like substance. And you can see here under the electron microscope pictures. And we believe this glue increases the surface area of contact between the pad and the substrate. The way the root hairs attach to the substrates is through what we call mechanical interlocking. If this one down here in yellow is the substrate, if we magnify, the substrate is not smooth, but is very complex and is composed by asperities and valleys between these asperities. When a root hair with the swellable tip arrive in this area, in this valley, the root hair swells, and once is swollen, mechanical interlocking occurs, and of course you have to think at the pad with all the digitations, so all this area of the substrates is covered by the digitation of the pad, and mechanical interlocking is provided, and it's very strong. Another way to see mechanical interlocking is in this graph. Here you see the root growing on the surface of the substrate. You see here are the valley between asperities, and root hairs penetrate inside the valley and are surrounded by the glue-like substance, which increase the contact with the substrate. When we look at the travertine instead of the calcarenitic stones, something else occurs. Together with the attachment of root hairs, there are, in the travertine blocks, there are several little holes. And when the tip of the root, in this case, happen to get in contact with one of these holes, the tip enter inside the hole, swells and become tough. And this provides a further source of adhesion. We wanted to measure the strength of this adhesion, and we did in 3 different ways. First, we used a pooling test to see the strength of adhesion of the seedlings, and we measured a strength ranging between 4 and 30 newtons. 30 newtons is 3 kilos. Then we put seedlings on a flume, and we saw that seedlings were able to withstand a linear current velocity of 70 centimeters per second for 40 minutes and an orbital velocity of 25 meters per second for 18 hours without detaching. And these are pretty strong currents and waves simulation. And this is just a short video to show you when the seedling is attached to the block, you can lift this block weighs more than 2.5 kilos, and so the attachment is really, really strong. Another thing we wanted to assess was whether roughness or if there was a roughness particularly suitable for guarantee a very strong adhesion of root hairs. So we constructed resin tails, these little things here, with different degrees of roughness. We used a peeling test to assess the strength of adhesion. We recorded no adhesion on glass, but we recorded adhesion on all the other degree of roughness. However, the greatest attachment occurred at roughness level of 12 microns. So with all these ideas, with all these results, we decided to construct what we called the perfect microsite. This is a stone, is obtained through a stone. In this case, it's a calcarenitic stone, but could be a travertine, because these stones have a suitable roughness to optimize root hair adhesion and interlocking. There is a slot identified here with the angle alpha to allow rhizome growth and mechanical interlocking of the rhizome in this case. And there is a small crevice down here, down in the slot, to facilitate root expansion and mechanical interlocking of the root. Another important phase after retention and anchorage is establishment. Establishment begins when retention and anchorage translate into survival, rooting, and growth, and represent the transition from being held in place to becoming an independent growing plant. And there are quite a few examples of establishment after transplantation, but I would like to show what we did with seedlings in Favignana Island. In this island, before the area was protected, both produced a lot of damages to the meadows through anchoring. And there are lots of scars like this one on the meadows. So we wanted to use seedlings trying to stitching up these scars. In this case, we used pot filled with stones because at the time when this experiment started, the perfect microsite was not already ready. So we let grow for a few months seedlings inside the pot in tanks. Then we moved into grids at different densities, and we moved the grids into 2 sites characterized by dead mat. And one of the sites had a thin layer of sand, and the other had not this thin layer of sand. After 2 years, the seedlings became little plantlets, looking pretty happy. And after 5 years, when we stopped the experiment and removed all the grids, the plantlet became pretty big. The results of this study say that in the best site, the one with the thin layer of sand, at the best density, which was the intermediate density, percentage of survival reached 75%, and the increase of rhizome was 200%. One important thing after establishment is expansion. Especially in restoration trials, it's a practical sign of establishment. And we can assess this establishment when a shoot or a rhizome not only persist, but expand behind the original planting unit. Could be a frame, could be a pot. And in the experiment of Favignana, this is what exactly happened. Retention was assured by the pot where we put the seedlings. Adhesion was assured by the presence of stones inside the pot, and root hairs made their job. But then we had establishment when the seedlings started to grow a plagiotropic rhizome and more orthotropic rhizome, and went out of the pot and started to colonize the surrounding habitat, producing very, very long roots. What we are doing next is trying to use this module, the so-called perfect microsite, and there are a couple of experiments at sea already running. So in conclusion, I'll try to go fast. As we know, Posidonia oceanica can recover naturally or through active restoration. However, it is important to deepen our knowledge on processes such as retention, anchorage, and establishment, as we saw during the previous talk. In a restoration project, it's also important to me to study how and if transplanted propagules expand outside the frame, the pot, or other devices we use to fix them to the substrate. So, as a recommendation in the context of restoration, it will be important to assess the nature of the surrounding substrates, as this may determine whether transplants can expand beyond the initial planting point. And that's it for my side. Thank you very much for your attention.
Thank you. Thank you very much.
Thank you. Okay, before going to lunch, now we have the second session. Trying to answer. I ask you the presenters to be aware about the questions. The first one is for Frederick, that is here in person. Frederick, could you use these results as a prediction modeling for future areas of Posidonia meadows in danger due to the increase in sea temperature?
Could you use this result as a prediction modeling for future areas? Maybe, but I would not be able to tell you how that exactly works.
Yeah, it's difficult to answer.
No?
This one is also for you. I might have lost some info about the experiment, but are you trying to induce flowering in seedlings?
That's what we're going to do in September, hopefully.
Yeah.
Yes.
Yeah, it's trying. For Albert, can you use on-field Posidonia points and then combine with AI and satellite images to have a more precise mapping of seagrass meadows?
Well, yes, like anytime you have like in situ point, it's better than anything you detect remotely. So if we had in situ points of the Posidonia like limits, you could use it to correct your detection or as a ground truth for training, or for example, for better defining the silhouette for the post-processing. So yeah.
But the question was because the
those points are not like are points that you see in the image that there's Posidonia, and when you see how the Posidonia evolves, there's still Posidonia there. No, more points of that kind doesn't lead to a better detection. Normally, it's better to keep it at minimum, and the minimum that the artificial intelligence better detects the limits, because if not, you're just marking the limits, and it will always stay inside the limits that you mark.
Yeah. Okay. Yeah. Thank you. Now for Alex, do you have in mind to try in places with different sea conditions, how could you control the landing area in places with currents?
Nowadays, I don't think so. We don't have in mind to try it in places with different sea conditions, because as I said, now we are trying to obtain more information about the development of the fossil rocks in the seabed. And for the second question, how we would control the landing area, it's difficult in current conditions, but maybe this could be a second phase or second stage for the experiment to control this type of situation.
Yeah, thank you, Alex. Now for Fabio, very interesting study. Did you make your blog, or are they commercially available?
Yes, we did on purpose. To be honest, we did 3 different kind of blogs to assess which one was the best to perform seedling adhesion, and we spent lots of time thinking at the structure of these blogs. So no, they are not commercially available, but they can be reproduced quite easily.
Okay. Thank you, Fabio. To Albert now, how easy and fast could it be to expand your work to other areas?
So it would be pretty easy, because you would just need, as Sentinel has cover of all Earth, you would just need to put the coordinates and then look a little bit at the image to see where you're going to put your ground truth points, and then you just let the code run, and it would take 1 or 2 days because it has to download a lot of images, but yeah, pretty easy.
Thank you. For Patricia, this is an interesting study. Could the 2 sites differ in environmental factors other than temperature?
Yeah, for sure, the hydrodynamic conditions. And then we were checking the salinity, we were checking the chlorophyll. There were not so many differences, but for sure there are some other many environmental factors that we didn't consider.
Okay. And for Fabio, how could anthropic artificial breakwaters barriers affect the proliferation of seabeds?
Well, this depends a lot on what kind of breakwater have been deployed. If they do not have any crevice or holes, any way to allow retention, I bet there is no chance to proliferation of the seabed. But also depends on the depth at which the breakwater are deployed, and also on the site, if it's a site where Posidonia used to grow or not. So in theory, yes, they could help. In practice, we should look at several things, the one I said before.
Okay, there is a comment from Andrea that said that I know that in some sites in Italy, some concrete structures have been laid. Yeah, it means some artificial reefs. And there is now question for Alex. How can seedlings flower?
Well, we don't really know that, but we may think that if they can develop in the seedlings, they can develop in the seabed. When it happened an event of flowering in the Posidonia meadow, it can be a possibility that these seedlings could flower.
Mm-hmm. And for Fabio, where we can find more information about your experiment?
Yes, most of the data I reported have been published. So if you write me an email, I can send happily papers. Of course, in this presentation, there are photographs you may not find in the papers, or few details you cannot find. But this is because, as you know, reviewers quite often say, oh, you cannot say that without the right data or other things. But most of the data I've shown today are found in published papers.
There is a comment for Frederick. Is this very unlikely? Seedlings can develop successful flowers. Even if they flower, many will abort because of the energetic cost of fruiting.
Okay, thank you, Fabio.
Okay. Well, is there any other question here? People in person? Yeah.
We are working with Lawrence Thompson. That's possibly for Fabio, but also for the guys, that are developing the badminton method, in which you attach the plant and you drop it from the boat. And Lawrence Thompson is, that you know perfectly, he works in University of Gothenburg and he developed the crawlers. So we have more than 15 years working with the crawler, with the robotic arm. So we're thinking to import precision agriculture methodologies to seed Posidonia and other stuff. And my question is for you guys is that would it be possible to, let's say, make holes into the, let's say, reefs, carbonate, whatever rock is around, and put the seed inside the hole? Would this allow some sort of flowering? Would that be possible, in your opinion, to like seeding Posidonia into holes in the ground or in the rock?
So if you produce, you produce a crevice or
yeah. When you plant the rice, there are machines that may do holes and insert the seed directly into the hole. That's something that we're working with Lawrence. Would it be something feasible? Everyone ever tried to seed the Posidonia making holes into the substrate?
I'm not listening from home. There is an echo, so I'm not sure I've perfectly understood. The question was whether it's possible to directly put seeds in the crevices underwater.
Making holes on the seabed through robotic means to put the seeds.
Okay. In theory, yes. Why not? What I observed underwater was a kind of herbivory on seeds. So you should take into account that seeds can be eaten by invertebrates, I think. I have no idea who was biting the seeds. But yes, why not? If retention is provided, and you should construct a perfect hole or a perfect crevice, then the seedling will all of a sudden develop root hair and start to grow. You should find the right place, and the right place is a place where Posidonia was living in the past. It shouldn't be a new place for Posidonia. This is if I well understood your question.
Thank you. Yes, thank you very much.
I wanted to add something.
Oh, yeah.
Hello.
Yeah.
Some colleagues tried to plant Posidonia seeds just like they do with Zostera or with Cimodosia. Consider that different seagrass species behave very, very differently. And for example, Posidonia seeds are photosynthetically active. So that means they prefer light. That's why they grow better on a hard substrate where they can attach rather than inside the sediment, because the seed usually naturally does not bury. They try to do that in dead mud, and because of the anoxic sediment and because of herbivory, the moment there was some leaf going out of the sediment and someone took it, it's not that they ate the whole plant, but they might take the seedling away, and that difficults a lot survival. So I think it's more feasible to drop seeds in rocky or hard substrate or dead mud rather than planting them inside the sediment, by what some colleagues have found.
Thank you.
Thank you, Fabio. Frederick, you want to do something?
No, that's it.
Okay, this one. Okay. Yeah, please.
So for Albert?
Yes.
I really enjoyed your presentation. I thought it was really interesting the way you use AI to map that. I was wondering throughout your presentation you consider perhaps the use of ROVs in order to map the area, and that might help you feed in extra information into the AI model that would help you get past the depth issue.
Yes, I have. Like, there's a lot of useful tools you can use to have, and not only like picture, but they would possibly be better. Now we're trying to have systems for that. So for example, with front of your imagery, because it would be way more precise. However, the thing about Sentinel is that you will just have it there constantly. So with that and satellite imagery, you can have constant output of data without having to go on a campaign to make data. So the perfect scenario would be having constant data from Sentinel, and from time to time you go out on a campaign, get data from LBS imagery or something like that, which gives you more better, more precise detection, and you can use that to correct the detections from satellite. That would be an ideal case. But yes,
Okay, then let's go for the lunch break. Okay, thank you for the presenter.
Right. Yes, we can start. Thank you very much, everyone, for coming back after lunchtime. We've got a number of interesting talks. The first one's going to be presented by Gigi Torres. She's a marine biologist and scientific advisor, a diving instructor with a strong focus on research, conservation, and restoration in marine ecosystems. Her career began with specialization in coral reefs and tropical ecosystems, and later evolved towards seagrass meadows and conservation of biodiversity in the Mediterranean. Since 2018, she's been a partner and director of Albatross Diving, proud to be the first PADI Eco Center in Mallorca. She's active in Balearic Diving Centers Association with Mare Baleare and is co-founder of Daughters of the Deep, working to close the gender gap in the marine industry.
Thank you. Yes, give me 1 second. Good afternoon, everybody. I hope I'm not going to make you guys sleep through my presentation. It's the worst time slot possible after lunch. So bear with me, we're going to keep this interesting, okay? We've had amazing, strong scientific talks given to you guys before. And from what I've seen in the program, I'm opening the more community-based block of the afternoon, which I'm really happy about. Let's not forget, we need strong science to make a change, but us scientists are very little strange people out there in the world, and without the help and the assistance of the community, we will not be able to make a change that will last through time. We don't have the resources, not time, not hands, not money, but most of all, if we're not able to change the perspective of people, all our conservation strategies will be lost and they will not be able to persist in time. So today I'm actually going to talk to you guys about 2 case studies we have. I come from a non-for-profit, first non-for-profit of the day. I'm based up in Mallorca, in the north side. We are Arras Marinas, a small local non-for-profit generated by local people for local people. And the idea is we try to do science that is locally engagement, and through environmental education, make a change in our community that will actually look after our marine environment for future generations. And when we talk about future generations, it's us when we become old, and it's our kids and our grandkids. And we want them to be able to enjoy the beautiful sea that you guys all work to preserve. We have done a community-rooted seagrass restoration project in which we highly involve the community in the restoration in 2 areas of Mallorca. So for those that are not very familiar with Mallorca, we're out here in the Balearic, and we have got, up in the north, we have got Alcanada in the Bay of Alcudia, and Canamurta in the Bay of Pollença. We do have a main motivation, which is to engage our community as a long-term solution for marine ecosystem restoration. But we cannot do just out of the will of making the perspective of people change the perspective of people. We need to have a solid scientific base to generate data that will actually prove that what we are doing is worth it. Yeah, because as we all know, without data, we have nothing to prove. So we are looking to evaluate the efficiency of restoration techniques, which I'll talk to you guys in a little bit, but we've tested 4 different techniques in our restoration projects. We're looking also at identifying the optimal transplantation season, as well as assess the biodiversity, biomass, and a bit of blue carbon recovery. So moving into that direction as well, because if we don't talk about money, there's no words going into the policymakers' ears. And also promoting that long-term coastal resilience through citizen participation. Right. As mentioned, we started in 2023 in Alcanada, and as a scientist, I believe we had to do a very scientifically solid base study in which we could bring our community in, because if they see that the work that they are doing actually has valid results and that we can later publish something that the scientific community can look at, they will make it theirs. And once a community makes a project theirs, you don't have to tell them to come and join you. They actually go and tell their fellow people in the community to go and actually participate in these projects. What we did, many of you guys have already talked about widely used restoration techniques, like the iron reeds here. But we decided, hey, let's not just replicate what has already been done, because we already have the results of this. Let's try and innovate a little bit. So we brought in 2 techniques that have been previously used, the iron rod and the cane, okay? But then we worked with a local artist, a sculptor, and he designed 2 degradable structures made from local material. Somebody before, who was the last person from the morning session, Fabio, he talked about with the seedlings, with using local stone from Italy. Well, we did something similar using mares, which is sandstone. That's what our Mallorca is made mainly. And he designed this anchoring technique, okay, which is, we named it mares. And then with terracotta, yeah, basically what our tiles are made out of, we made sure that it's just terracotta, using a similar strategy to just pin it down like staples, like gardening staples, but using natural degradable material. We then decided to establish 4 replicates using same techniques that have been used before, meter square, 20 fragments in each plot, and 4 replicate. And we headed out and chose an area that's easily accessible by the community, that it has a depth that people can actually participate in the process. So we did at a 6-meter depth. And before actually establishing the project, we made sure that that prairie that had been degraded by environmental factors did not have those environmental stressors in them anymore, because why are we going to be restoring something that is going to continue to be degraded? All our efforts are just going to go out the bin. We determined that area, which is fantastic. I really encourage you guys, if you are on social media, check out Aras Marinas. You can see all the videos, the details of how this is done. And when we started with our project, we had over 100 volunteers join us throughout the different campaigns. They help us collect the fragments. So we also talking about stress factors. We collect the fragments the same day and replant them the same day. So we have a team, head out, collect the fragments, and then there's land-based team. So again, making sure everybody can participate. You don't need to dive. You can help us with measuring the length, getting all those factors that are really important to be able to do the follow-up. And then out plot by plot to replant, and we did not collect any more fragments than that we could not plant the same day. Of course, we made sure to use all those variables that are already established: minimum length, minimum number of shoots, healthy young roots, okay? And so we headed out, we did all the replanting, and for the first 6 months, we went out every month and did a follow-up of these fragments. Follow-up at the start consisted in counting the shoots and everything, but it got to a point where there is quite a bit of sedimentation in the area, and we found that if we continued to look through the shoots, we were actually generating more disturbance in those fragments that are trying to root down and helping them. So after the first 6 months, we started to go every 3 months, and after the first year, we go every 6 months, and now we go once a year to do a monitoring technique, which actually we just headed out a couple of weeks ago to have a look, and it's actually really inspiring to see people that started this project 3 years ago, that they call it their project, and the sense of belonging that they have. And they go home and they tell everybody about it, and it changes the perspective towards seagrass. Because, I don't know, are many of you guys from a coastal area that has Posidonia? Raise your hand. Okay, good. Now I have a question. How do people call this? A seagrass or an algae? Okay, an algae. It's that los algares. We don't want that. We need to start changing that. They need to understand that thanks to this, we have the beautiful water quality that we can enjoy. It produces oxygen, and most important, it is our biggest ally towards climate change, to natural-based solutions through climate change. And thanks to a project where people can get their hands wet and dirty, we're starting to change that perspective. But then we said, hey, let's not just stay with one site. Let's go out there and let's replicate it in another area. Because, okay, we got good results. These are our results so far. We're actually having around an 85% survival rate in the iron, which is this one. We'll later discuss. I'd love to hear everybody's ideas with the different techniques. The reed, we got less than the 60%. Of course, it is super degradable, very mobile. It's just 2 bamboo sticks in the sediment. And then terracotta mares have around 70 to 80% survival rate, which is fantastic. But we're not satisfied with just one site. If it worked in an area that had high sedimentation, let's go check out what happens in an area where there's low sedimentation and a bit deeper. We headed out and we were looking for a bit of literature, and it's always the same as with regards to seasonality. It's great to plant during April, in spring, but we want knowledge. Let's get our community involved to actually test. Yeah, so we've done a replanting in summer, in autumn, in winter, and spring. So that we just finished last week with our last replanting, and it has, in the first project in Okinawa, we planted over 320 fragments, and now we've just replanted over 500 fragments collected by hand every day that we go replant. Next. Hey, there we go. So basically, we believe that a social-ecological framework which combines strong scientific evidence with a high community engagement, it is going to be the solution to change our mindsets in the future. Yes, but if we actually have a look, we cannot talk about coastal resilience if we don't talk about environmental education, not just for the little ones in school, which luckily is starting to be implemented, but also for us, for our colleagues. How many of you guys go have a beer at the bar that tell you, you're too geeky, I don't understand what you're telling me? No, it's okay. It's okay to be part of the conversation. It's okay to spark that interest. Community has to involve, but above all, if we don't do a change that has a strong scientific base, we will not get to the top of the pyramid with coastal resilience, which is what we're all looking for. But we don't stop here. We're looking for doing a full biodiversity and blue carbon assessment, check out if restoring through replanting actually has a positive impact with the blue carbon absorption. We're looking to expand to new sites as well as integrate to climate adaptation strategies. But thank you very much, guys, and thank you for staying awake. And I really hope that I spark a little sense of making sure that those next to you are part of the change and part of the conversation. Thank you very much.
Thank you, Gigi. Our next talk is by Paul Aguiló. There you go. Paul Aguiló is a professional at the Centre de Estudis de Mar in Sitges. He's part of the Barcelona Provincial Council, where he works in marine environmental education, citizen science, outreach, and marine conservation. Focuses on bringing ocean literacy closer to the public and promoting environmental awareness, stand out for his continued sustainability.
Well, good afternoon. Thank you, Frederic. And thank you to the organizers to give me this to share with you the project we are doing to monitoring and the state and evolution of the Posidonia. We have Vilanova using a science methodology.
Sorry, just a moment.
Yes. Well, English level is to pass it, so I do my best. And I hope I will be able to give you a general idea what I am doing. So well, let's go. Well, this is the list of contents that I would like to share with you. First of all, brief introduction about where I am working, what the Marine Study Center is, only to contextualize our action. The second point, a brief explanation about the environmental value of the marine area, the Geltrú marine area, and a brief description of the Siglos. And finally, the presentation of the citizen science project, description, methodology, the results. Well, the Central Studies del Mar, it's a marine environmental education facility. What we are doing is work with students, primary and secondary school, and families, citizens as well, to give them knowledge about the importance and the state of the ecosystems and its biodiversity. We are focused our dissemination in Mediterranean Sea, in concrete in the marine area. And that means that we are not scientists. And we are working since the point of view of education with the environmental education. Finally, that we want is to give people the opportunity to participate in projects to conserve the values of the marine ecosystems. And this is the Geltrú marine area. It contains from the mouth of the river Llobregat until Riufoix, the mouth of the Riufoix, line of the coast, until 50 meters depth. That means 26,000 hectares of a marine area. And this marine area, it's recognized that Natura 2000 network. And that means that at the European level, is recognized its importance, its biological and environmental importance, basically by the existence of this key habit, like is the Posidonia oceanica. And also the existence of some characteristic species like cetaceans, dolphins and fin whales, seabirds, and careta. The dominant marine current in this area is from north to south, And here it's important to see that in a very short space, we can find a lot of ports and infrastructures that are changing, that are putting barriers to the natural sedimentation dynamics. For example, here we have the port of Barcelona, Aeropuerto del Prat, where also we have Port of Alcarca, Port of Aguadols, Port of Garraf, Port of Vilanova and Geltrú. So in only this space, we have a lot of ports and infrastructures. And this alteration of the natural sedimentation dynamics not only affects to the sand of the beaches, also we think that it's affecting the sedimentation of the seagrass meadows. The seagrass meadows of Garraf marine area, it's about 400 hectares. It's a habitat integration due to sediment dynamics, rising sea temperatures, and other anthropogenic impacts. I think that we can say in general in all the Posidonia meadows of the Mediterranean Sea. And it shows very degradation in this area, and in a better way, density and coverture in this zone, from Vilanova to Huellas. And this Posidonia meadow is located about 16 to 20 meters, more or less. And close to Huellas, we can find Posidonia in about 10 meters as well. Okay. At this point, it's interesting to see that in 2025, the government of Catalonia published a map of marine habits. And if this map is compared with the map we had in 2011, we can observe a great regression and alarming regression, I think. But we cannot, I cannot tell you an exact data, an exact figure, because we are not conducted in a depth analysis of the methodologies used in each of these mappings. But I think that this is a point of interest that we have to resolve. And another project, this is a citizen science project. That means that we are working to get our data with scuba divers volunteers. We are working with about 30 scuba divers in total. That means that one of them are collaborating, and in other dives are collaborating other divers, but always this group of 30, more or less. So we are giving them training. They help us to get the data we need or we want to achieve. And we are promoting this project with the collaboration of the Federation of Underwater Activities, FECTAS. And we have two places of study, two study stations. One of them is located at 18 meters of depth, and the other one is located at 16 meters of depth, more or less. The reports that we are generating are shared in our project website, Science 2024. So in our website, we have two reports, 24 and 25. But we started the project at 23, but in 23, the objective wasn't to get data, but the objective was to put the two study stations in order. And what we are doing with divers, well, first of all, we are giving them training And we are organizing two different kinds of divings. One of them is to get the biological parameters, seagrass density, seagrass cover, plant burial, and sediment dynamics. In future, we think that we will be able to observe if exists regression and expansion, because every study station is marked with 5 iron marks that are located in the boundary of the seagrass when the seagrass is ending and starting the sand in the seabed. Okay, so we can in future, I say in future because in 2 years the changes in this sense are minimum. No? But in future, maybe we will be able to see if the seagrass is more far than the market, than the iron market. So one of the type of divings that we are doing is to collect this data, and the other one is to get or to see some biological indicators like its flowering, and to take photographs of the biodiversity associated in this marine area in the seagrass. All of these photographies we identify the species, and we share on the Minkia Citizen Science platform that is promoted by Institut de Ciències del Mar from CSIC. Some results, only 2 years of data, so we are not in this position to give solid information about the evolution of the seagrass. But we can see, for example, that the station 1, it's more deeper, has better results, stable density and cover. And for the other hand, the station 2 is shallower, 16 meters, shows signs of regression. This is an hypothesis we have. Again, regression linked to the sediment dynamics, rising sea temperatures, and other anthropogenic impacts. Now, we are not in this position to go more in time. About the biodiversity, we have detected 164 species in 12 dives with 2 volunteers. For us, this is a good number of identification. We think that it could mean that the meadow remains a high value ecological area. Conclusions. We have observed by the map of the regional government, and also looking the cover tour of some points of the meadow, we think that it's obviously that this Posidonia seagrass is in regression. We would like for the next year to have a third study station close to Cubellas and at minimum depth possible, because in that way we will have 3 points in all the superficie of the Posidonia meadow in a different depth, and we think that in that way we can enrich our analysis. And the more, or a point important to think about, if the citizen science is good or not methodology to work this kind of projects. Well, obviously, it's necessary to explain or to think how we can give better instructions to the scuba divers volunteers. And in this sense, for example, at the time to get photographs and identify species, I think that this is a very good methodology, because you can observe all that you see, or if you have some instructions. You can focus your observation in one point if you want. I think it's something very easy for the volunteers to develop. On the other hand, to get the data about biological parameters, sometimes could be more difficult because it's possible that the data recollected it's not exactly right. But also, we are working with a limited number of scuba divers, and train by training, we are improving the way we are getting the data. And on the other hand, maybe the results in this sense are not 100% 100% fiable, but I think can show the dynamic of the changes or the evolution of the seagrass. And in this sense, it's also very important, the money. The money is important in our life and also in the citizen science, because we are paying or we are financing this project year per year. So we don't have a perspective about we can develop that next, I don't know, 5-10 years. And this is important because it's difficult in these conditions to tackle the points of improvement that you detect. In fact, this is the main objective that we have for this year, to try to clarify if we get the way to give a perspective of how many years we can develop that, to give a planification of improvement, or to go more in detail in some aspects and others, or to put another study station, like we would like. Basically, this is all that I would like to share with you. I hope you enjoy, and thank you.
Next talk is with Marcia Cianflone. How do you pronounce it? Cianflone. Is a PhD student in marine biology with a strong specialization in remote sensing applications for marine ecosystems. Her research focuses on analysis of satellite imagery to monitor coastal and benthic habitats, with particular attention to Posidonia oceanica.
No, sorry. No, thank you. Thank you for the introduction. I'm so glad to be here, and because also restoration was something that wasn't my research as an objective, but I think that satellite remote sensing could be really useful to help the restoration of Posidonia oceanica and other series. So before starting talking about restoration, as a biologist, I couldn't start talking about some general information of the importance of the Posidonia oceanica. It is an endemic species of the Mediterranean Sea, so it's nowhere else present in the world. And it's a really important species because thanks to its structure, it is an habitat former, really important for many other species. So we can say that Posidonia oceanica can really support the biodiversity of the Mediterranean Sea. And also because it is a plant, it can produce oxygen, and it also works as a carbon sink. And we all know how many ecological importance roles it has. But because it's a shallow water habitat, it's also really sensitive to the changes of the environment. So we know that the main causes of the disappearing or reduction of the Posidonia are like the poor water quality, the construction of coastal infrastructures and other human impacts like anchoring, trawling, and a lot of factors that altogether are causing the declining of this habitat. So here comes the restoration, because sometimes we know that the only protection of the habitat is not enough, because there are some areas that cannot be protected enough, and so restoration could be really useful to help this habitat to recover. And we know that there are two kinds of restoration. The one that is called natural or passive, because with the mitigation of the human impact, we can allow the natural recovery of the habitat. And on the other hand, we have the active restoration, that means actions that directly help the recovery and the reconstruction of the habitat. I don't want to go deep the different methods and techniques, because you are more expert than me, but one of the main criteria that we must follow before doing our restoration is the choosing of the site. Because we know that now one of the most important drivers of the success of the restoration is the correct choice of the site. And here are some works, for example, the one of Arianna that she explained this morning. And so what satellite can do is to help this decision. Here other works about the macroalgae habitat, because it's the same for also other kind of species, that the historical presence of the species in the site that we want to restore is one of the fundamental drivers for a successful restoration. Satellite remote sensing, as also my colleague explained before, it's a technique that acquires the light reflected by the Earth's surface. And in shallow water, the light can penetrate the water, can reach the seabed, and depending on the type of the seabed, the light is reflected differently. So with thanks to a lot of satellite sensors, we can have different kind of signals and can reconstruct the habitat and make some maps. Another quality of satellite is that we have a lot of archives that are really long, go back in time. In this table, I summarize some of the characteristics of the main satellite used for Earth observation. We have the cost. Some of them are free, open source. Others have really high cost. There are different spatial resolution, of course, and different coverage, different daily, different revisit time. And here in the column that I highlighted is the year of the start of archiving, acquiring Earth images. So we can see that altogether we have like more than 30 years of acquisition that we can use to monitor also back in time our habitats. For my PhD, I developed an algorithm. I am a biologist, so together with my supervisor, we developed an algorithm to do this kind of monitoring of the Posidonia oceanica. What I want to say is that studying from literature, we observe that there is a lack of information about the method of using satellite in marine habitats. So what we wanted to do is to create a workflow that it can explain well all the steps that must be done to do this kind of monitoring. Because at the moment there is no protocol, no standard protocols about. And the objective of this workflow is to obtain at the end a map of the change of the Posidonia oceanica meadow, because we know that to map the Posidonia oceanica meadow, there are many other technologies that have a really high resolution, but thanks to satellite, we can have a more global spatial coverage, and also we can go back in time. And we want to see how the Posidonia oceanica evolved in time. I will explain to you some just steps. I don't want to go deep in the method, but here is how the archive of one of the satellite PlanetScope, how it looks. And I choose many images from this archive. It is a daily revisit time, so we have really a lot of image that we can choose. Of course, we choose the one with the best atmosphere condition. And here are two examples of the Posidonia oceanica here in Vilanova and Geltrú. The first image was acquired in 2019, and the second in 2020. So what I've done after collecting this image was to compare all these images and see what changes in terms of the signal, the radiometric signal. First of all, after processing all these images, I used in situ data. In this case, was the marine habitat map provided by the Generalitat de Catalunya to do supervised classification of the habitat. This is so important to train the algorithm to do itself automatically this kind of classification. So I just have to specify that this kind of map, as someone of you already told before, is from 2024. So, of course, it's classification asynchronous, because those images are from here before this kind of map, but was useful in any case. And on the right, you can see the result from the algorithm. Each dots are is a pixel who was classified with a high confidence, only with high confidence. In green, there are the Posidonia oceanica, that of course. And so I compared these two, but also other images together, and obtain this map. That is the map of all the changes that the algorithm obtain after this comparison. Of course, there are many false positive, other changes that could be temporary changes, and we filtered this only with taking only the pixel that regards the Posidonia oceanica meadow. Despite the color, I'm so sorry I have to change it, it's a loss of Posidonia oceanica. It's not a gain of the meadow. And we are not sure at the moment, but we know that in 2020 there was a big storm that could be the main cause of this loss of Posidonia oceanica meadow. But what I want to tell now is that after all this, we can use satellite to analyze and to see if there are any areas in which there was a historical presence of the Posidonia oceanica meadow, especially maybe in areas in which we don't have any data from the past. So I really believe that this could be a really useful tool for restoration management, and to improve also the successfulness of the restoration. So thank you so much.
Thank you, Marcia. The next talk will be online by Eugenia Apostolaki. Eugenia Apostolaki is a senior researcher at the Institute of Oceanography in Greece. Is that right? Yes. Her research focuses on the ecology and biochemistry of seagrasses. She studies the response of seagrass to environmental disturbance and global change. and she is particularly interested in assessing the role of seagrass meadows in climate change mitigation and adaptation.
Evgenia, we can hear you. You have your microphone muted.
Yes, sorry. Hi, everybody.
Hello.
Can you see my screen?
Yes, we do.
Okay, thank you for inviting me. I may be a little bit out of context, I'm not so sure, because all the previous presentations were very technical, and I'm going to present you a more broader perspective about Posidonia restoration. So let's see. Of course, seagrass restoration is not a new concept or practice. The people have been working on restoration of Posidonia for many years. You can see relative information in older papers. However, the recent international policy developments, such as the GBF and our nature restoration regulation in Europe, have changed the way we do restoration, because they have established quantitative, time-bound, legally bound also targets that require quantifiable pre-restoration assessments and the use of harmonized indicators to monitor and report the progress of and the success of the restoration. So for Posidonia, we are very lucky. Because there has been already in place policies like the Habitats Directive already from 1992 and the Water Framework Directive and the Marine Strategy Framework Directive, that all these provide a lot of information about the extent and the status of Posidonia meadows. And this can give us what we need, like the reference values that we need for restoration, the assessment of the habitat damage and loss, and also the metrics to measure the potential positive outcome of restoration. Of course, the problem with these frameworks is that they rarely assess the key ecosystem services, let's say, other processes that are very important to understand the degradation of an ecosystem. At the same time, we are lucky with Posidonia because it's the blue carbon queen, as you know. And these restoration frameworks link the restoration to the blue carbon reserves and give an extra opportunity for people working in the Mediterranean and with these species to increase the efforts towards restoration. The problem here is that there is no policy in place, but we do have the EU climate law and the LULUCF regulation, which lately they allow for the reporting of wetlands, not for seagrass, but wetlands, and they give this window, let's say, to include the emissions and removals of Posidonia meadows to our national inventory. So these policies, despite their drawbacks, they can measure the structure and function of seagrass ecosystems and provide some foundation for standardized measurement of the restoration outcomes. So I will show you what we are doing in the frame of Artemis project along these lines that I already explained. So we selected a site in the eastern part of Crete. It's called the Tsikiari Bay, and we selected this site because we knew that there was a fish farming that operated for more than 20 years, causing excessive seagrass loss. But also it was removed 7 years ago before our interventions. And the meadow, what was left of the meadow, showed signs of natural recovery. Also, this is part of a Natura 2000 site that will receive management measures next year. What we did was here you see the map where with the shaded area is where the fish farm used to be. First, we mapped the meadow using drones and ROVs and scuba diving. And then we selected different stations to measure the ecological quality of the meadow and what is left of it. We took a course with blue for blue carbon, and we assessed biodiversity through standard techniques like quadrants and transects and cores and the DNA. Here you see the map with the brown. It's what is all dead mud. With some shoots here, there are some shoots very low cover. Here you see in the middle that it's mixed with dead mud, and the cover is medium. And here there are with the darker green are the patches of Posidonia. We collected samples to do, as I said, the ecological quality, and we used the national intercalibrated indicator, WPOSI, that takes into account the shoot length, the shoot density, the lower limit, and the rhizome number of horizontal rhizomes, and other indicators like conservation index and substitution index. And we also calculated the ecosystem-based assessment for Posidonia, the EBQI, which is the only ecosystem-based indicator that we have until now, although it has some limitations. Basically, it's not calibrated for the Eastern Mediterranean, but this is the only synthetic indicator that we have to move beyond the seagrass and assess the whole ecosystem. So we found that due to the fish farming, there was a 60%, more than 60% loss of Posidonia. We have 6.5 hectares of dead mud, and the ecological quality for station 4 was moderate and good for the high cover station, which are very local assessments. And overall, based on EBQI, the ecosystem condition was bad. What we saw regarding the biodiversity, it was a reorganization and homogenization actually of the community. And here with, you see the fish, and here you see the invertebrates associated with Posidonia. With red is the no to low cover areas, and with green is the medium to high cover. You see a clear separation of the invertebrates, macroinvertebrates, and some lower separation for the fish. The salmon showed a decrease for the invertebrates in orange and an increase in fish, but towards the no to low cover. But this was basically related to the fact that in the Eastern Mediterranean, and especially Crete, we have a lot of invasive species, a lot of fish that are invasive, or well, non-indigenous anyway. So what we saw there is that we actually lose the species, the fish associated to Posidonia in the dead mud, but we have also an increase in the non-indigenous in the dead mud, because non-indigenous species are more generalists, so they don't really care about what they eat when they are in the non-native range. So what we see here, the increase in diversity is driven in fish, is driven mostly by the loss of Posidonia associated species and the indigenous species. Then for blue carbon, here you see the 1 meter stocks in the sediment. This corresponds to the fish farming period, assessed through the lab dating. And here you see the stock in the living and in the litter. You can see it here in the magnification. So you saw obviously we lose all the carbon that is associated to the living compartment in the no cover area, and we have very low carbon stock in the low cover and higher in the high cover. Here, this difference here is due to the fact that we have a different hydrodynamic regime in the area. If you remember the map, here in the low cover, these stations are more sheltered compared to these stations that are exposed. So basically, we measured the hydrodynamic exposure to be higher in this area than this. And we know people who are actually working with blue carbon know well that this is very much related to the carbon that you actually measure in the sediments. This was also evident from the grain size analysis. Here is also the mean value of 1 meter per station. And you see here in the no cover and the low cover, the high percentage of silt, clay, and fine sand that indicate a low hydrodynamic exposure. However, when we analyze only the top 5 centimeters of sediment with carbon-13, and we run the mixing models to identify the different sources of carbon in these top centimeters of sediment, what we saw was a lower contribution of seagrass in the no cover and low cover stations compared to the high cover stations. And of course, the contribution of other sources like other macrophytes that colonize the dead mud, because there we have Anopheles stipulacea, we also have Caulerpa species, we also have Padina papa, we have other species that colonize the dead mud areas and contribute to the carbon sources. But basically what we see here is this weakening of blue carbon towards the sites that are, towards the stations that are more affected from the fish farming. After the baseline, we did a cleanup because unfortunately the fish farm, they abandoned the site. They are supposed to take all their material, but obviously they take only what is visible or what is expensive. So we arranged an underwater cleanup to clean the area, and after that we did a small transplantation, a small-scale transplantation activity. We selected 2 stations along the direction, let's say, of the area. We took cuttings from the meadow that it was in the good status here at the more exposed part of the bay, and we transplanted shoots using different densities, one higher density and lower density plots, using muds and staples. And here you see the results. So then we monitor this. This was done in May 2025, and we monitor this every 3 months. Now this May was 1 year monitoring. Here you see the survival rates of biodegradable muds and staples until November. Unfortunately, we didn't have the time to assess the new data. And here the photogrammetry. So what we are doing now is taking water samples to measure the fluxes of carbon in the area, and also we collect sediment to assess the potential enhancement of biodiversity using eDNA. So we are here now. In this baseline, here you see the overall result of the project so far. We have the loss of the habitat, the ecosystem condition, the fish, and the invertebrates, the megagrams of carbon upscale. This is upscale carbon, and the megagrams of carbon per year. And now we have the project, the Artemis project is finishing now in June 2026. And fortunately, we have another project that will give us the opportunity to follow the monitoring of the meadow for a few more years. And potentially assess the additionality following the interventions. Thank you very much. I only have one request because I have to leave earlier at around half an hour from now. Is it possible if anybody has a question to do it now? Sorry for this. how did we calculate the fish when we transect? It's a typical assessment of fish species. Is this the design this study? Is this for me? How you design the study stations? So, I cannot hear anything.
Remember that in the room you are muted.
Sorry, Daniel, we were muted.
Okay.
Yeah, there was one question only, the one you read before. So I guess you replied.
Okay.
So no more questions from the hall? Everything was pretty clear?
Thank you very much.
Thank you very much for your presentation. Thank you.
Right. This one?
The next one.
Okay, next one. Our next speaker is Luis Stagnetto. Luis is a Gibraltar-born marine biologist who has dedicated his career to understanding and protecting the marine ecosystem in the Strait of Gibraltar. In 2016, he co-founded the Nautilus Projects, a local NGO committed to raising awareness of the marine environment. He completed a Master of Philosophy at Heriot-Watt University in 2025, focusing on phytoplankton cycles and structure.
Right, so good afternoon, everybody. It's a great pleasure to be here, especially coming all the way from Gibraltar to explain the work that we're doing and why we feel that this work is important. I think it's important for me to actually declare that we're working with our partners, Peninsula. And Peninsula are actually a bunkering company who not directly, but the port activities is what has eliminated our Posidonia beds and seagrass beds. So the point that I'd like to make here, like Gigi was highlighting before, is that by working and educating people, you can actually start getting industry to put money into this restoration, because this is the way that we are doing it, right? So Gibraltar and the Straits is a particularly special bit of habitat. It is the entrance of the Atlantic and the meeting point of the Mediterranean with that. And so we've got two very different chemical signatures within the water: high salinity, warm Mediterranean water meeting with cooler, fresher Atlantic water. And also biologically, it forms like the southern limit of Zostera marina, but also the western limit of Posidonia oceanica. So we have that overlap there. And both species, according to the literature, have been present along the Gibraltar coastline at one point or another. Unfortunately, Bull et al., whose study came out in 2010, but actually did the study in 2008, found that by 2008, 100% of our seagrass meadows were gone. And subsequent studies have not been able to find them either. So to give you a timeline, because I'm not sure how familiar you are with the area, but the first losses began in about the 1890s. And the reason for that is the British started establishing a port area with defined moles, and these moles literally went straight over the seagrass beds because it wasn't about the biology; it was simply about what was neat and what could be built. By the 1970s and 1980s, Gibraltar was running out of space in terms of places for people to live. And, it galls me to say it, but a whole area of Posidonia bed was literally just filled in order to make houses for people. And again, this comes back to this idea of if you don't know the value of something, then you're less likely to preserve it. You don't realize the significance of that particular species. And had we known today, what had they known in the 1970s and 80s, what we know today about the importance of Posidonia, perhaps we would have averted this. 1993, Shaw does a study in the area where the seagrasses are still present at that time. And like I said, 2008, that's when the complete extinction happened. So where we sit today, 2025, 2026, is we are looking at restoring some of those seagrass beds to the area. I think it's part of our marine heritage, and it's important that we re-establish that. And we're looking at doing that with Posidonia oceanica. So a number of speakers have already gone through the biology. It's late in the afternoon, and I'm not going to put you all through that. But our solution to dealing with this is a land-based recirculating nursery. Why? Ultimately, because we can control the conditions that are there, and that actually helps us to manage the stress of the plants and deal with things without having to put divers in the water. The other thing it allows us to do is it allows us to prepare the plants from one site, bringing them to the nursery, and we can graduate that those environmental conditions to the new environmental conditions that we want in the nursery, and that has to be of benefit. And it allows us to have very easy ability to monitor the plants as they are growing in the nursery, which when they're in the wild is just a little bit harder to control. A couple of Gibraltar specific hurdles. We don't have any seagrass left, so we don't have the ability to collect seeds. We don't have the ability to go and monitor natural seagrass beds. Every plant that dies in our nursery is a plant less that we've got in our stock, and originally we only got 100 plants, so we're a lot more limited. The western coast habitat, the bunkering activities still continue to happen in that area, so it's not as good as some of the other sites around the rock. Although again, this is where now, working with industry, we might be able to open up sections of that western habitat, pockets where industry doesn't work, and might allow us to use those. International donor sourcing, the plants have to travel a long way. It's not ideal. And it adds to the stress, it adds to the cost. We're working with Malta in order to force these grasses. And the ongoing urban pressure. Gibraltar's 2.5 square miles. There isn't a lot of space to build houses, and inevitably, whenever we need to do something new, we end up reclaiming land, and that has a tangible effect on the marine environment. So our project overview for phase 1. Our donor site was St. Thomas's Bay in Malta, and we worked with the government of Malta and the University of Malta in order to source the grasses. We followed a protocol that, because Nautilus formed part of the Mediterranean Posidonia network, we're a member of that, and they have some protocols for the cuttings, and we followed those protocols very, very carefully. The government of Malta actually put extra conditions on the way we were sourcing. So, for example, no more than 2 cuttings in every square meter. So we couldn't source from a Natura 2000. This stuff, we weren't allowed to dig out any parts of the plant. We actually had to get either the ones that had already been broken off the main fragment, or parts of the plant that were exposed. So these were the conditions that we were working under, and obviously the government of Malta were there to oversee how we were doing it. So 100 plants get brought to Gibraltar in September, the 30th of September 25, and those plants have roots and shoots intact. And then in our tanks, we started monitoring those plants as they were growing with whatever interventions were required. And at about 7.5 months, that's when we've done the first. So we've been collecting data throughout that time, but it's the first time that we've actually produced a full report on where we are today. Nursery system design, for those of you that we've got 20 tanks. The tanks are divided into groups A to E, and we had about 5 tanks per plant to try and lower the density. The lighting that we use, we have AI Hydra 64 HD lighting. They're really, really versatile, low power as well. The reason why I like them, because you can actually change the light profile. So what we've got is we don't use natural light. What we have is a light spectra, which is focused on the reds and the blues, which are the chlorophyll peaks. So we can actually make that not only that, but we can raise or lower the intensity of that light in order to simulate seasons. At the moment, what we've done is we've kept them at spring conditions. We're trying to get them to grow, we're trying to get them to establish themselves, but that's why we've got them at that level at the moment. Temperature, matching local conditions at depth, obviously because the surface temperature in Gibraltar changes quite a lot. So 16 degrees plus or minus 1, depending on the chiller. Salinity, 36. Why? Because of that interface. Mediterranean water is probably a little bit higher at about 38. Atlantic water is a bit lower at 32. It mixes, and the salinity around Gibraltar is about 36. So if we get plants used to a higher salinity or a lower salinity, when we put them out into the wild, we would have a problem. So that's why we've got them used to that range. pH, now, caveat on this, 8 to 8.5. It uses a system. We have a semi system, a reef system. So it's for aquarists, home aquarists, hobby aquarists. So it's not as accurate as it should be, and it uses a qualitative method of like looking at the color changes on a pad in order to assess that. So it's really hard to say exactly what the pH is. There's a little bit of a range of that. And ammonia, we got it at less 0.02 parts per million. So some of our headline results after 7.5 months. 82% of plants are showing signs of growth, not necessarily the right type of growth, and I'll explain that in a minute. But 82% are definitely alive. There's definitely some growth there. 51% have retained photosynthetic capacity, and of those, 21% are above the baseline. And what do I mean? Our baseline is that the plants have shoots and roots. Okay, so that is those are our main results for that. Looking at the total plants, so what we had when we got the plants in from St. Thomas's Bay, they arrived the same day. Over the next month or so, what happened was there was a big regression by the plants. All the leaves went brown. They looked like they had it, and then suddenly, after that first month, they started regrowing. Some of them at the top, the 18, they didn't, and all the organic tissue just basically started to die off, and so we knew that those were definitely dead. But we had a number of plants, so 40 plants, which were only growing roots. And this is interesting because they were clearly alive and they were clearly using the rhizome to continue to grow roots, but there was no sign of attempting to grow any shoots at all. Then we had plants that the root system just withered away, and or they went very black and woody, and they put all their effort into growing shoots. We had plants which had roots and shoots. They developed both of them at the same time. And the final category at the bottom was that in 7 months, these plants not only grew roots, not only grew shoots, but they actually showed signs of rhizome, showed signs. They were clear shoots coming out from the rhizome. There was clear rhizome growth. One of the interesting observations that we made at this point is that some of the plants from this root-only category suddenly, spontaneously started growing shoots. Nothing changed in the tank condition. It was, we put them in what we call recovery tanks. I'll go into that a little more. And they just suddenly started developing shoots. So that for us was an interesting observation. If we look at the system, what you see is that on a lot of these categories, the variance between the tank systems is practically the same, yeah? But there are two that aren't. And the first one is roots only. So in system A, the roots only, the purple, was significantly less than in any other system. The other category was roots, shoots, and rhizomes. So system A was the only system that actually started developing rhizome growth. And all the lights are the same, all the chillers are the same, all the conditions are the same. The sediment that we use is the same sediment from one thing. But there does appear to be a clear difference. Of course, it could be chance. It could just be random chance that's what happened. It's a small sample, it's 100 plants. But it is certainly an interesting observation. So our first key finding is that rhizome development was achieved, and this is important because this is not just survival. These plants are growing and are happy in the condition that they're in. Not happy. If a plant could be happy. Rhizome growth for us is a prerequisite before we can even consider either seeding, trying to seed them, or planting them out in the wild. So that for us is great because out of 100 plants, we've now got 7 that are definitely at that point after 7 months. And as we've seen in the various presentations, it's a slow-growing plant, right? It's probably going to take a couple of years before we get more of them over that line. Group A, whatever the conditions in Group A were, it's important to really drill down on that because I've had a lot of food for thought over the course of today, one of them being the scientist that was talking about the bacterial ecology within the sediment. And I suspect that if we analyze our sediment, we might find some sort of signature of the bacterial differences between the system. And that would be good because it means that before we put any more plants in there, or we can actually make sure that the bacterial community is what it should be. Our second key finding is that spontaneous re-sprouting. So for the transfer, we had the stress from moving them. We had these plants that were only growing roots, and in some cases we thought, well, all of these have failed. But this new shoot initiation is important because what we've done, instead of classifying those plants as dead and removing them from the system, we've actually got them sitting in the tanks. And we're slowly, in certain cases, getting plants back. So just because a plant looks dead doesn't necessarily mean that it is dead. If you keep it in the conditions long enough, you might actually start to see some recovery over time. And that, in our situation, where we've got such a limited amount of plants, is really, really important. So that's the thing. And the thinking behind this is obviously the rhizome. If there's sugars in the rhizome, the plant has the energy to invest in growing shoots. One of the thoughts that we've had is that it almost seems like some of the plants were genetically programmed to be dedicating their time into roots. And what I wanted to consider on this is that when cutting plants, if we could somehow identify a way to know whether a particular part of the rhizome is going to grow roots or shoots, we can actually improve the methodology of cuttings because we could identify potentially that this is a good part because it's going to grow shoots, and that means that it's going to have a higher survival rate later on. So perhaps what we've seen is it has some other basis for it, and if we could identify that basis, that would certainly be helpful. So the recovery tank, I did tell you, so C1 and C2 we ended up converting into recovery tanks. What's important about this, like I said, they have lower stock density, we have a higher monitoring rate on them. So we're going in there, we're checking the factors more often, we're checking the light with a par meter, making sure that they are all right. And the plants are not reclassified as dead until the viable rhizome tissue has completely degraded, rather than having roots and shoots. Why this matters? For us, every plant is irreplaceable. The recovery expands our outplanting pool, which is super important. And the protocol, in terms of that recovery, could be formalized a little more. So if you have plants that seem to be locked into producing roots, perhaps there is a mechanism that we can encourage them into growing shoots, and looking at the protocol to try and do that is something that we want to prioritize in the future. This has shown us as a project that nursery conditions is viable in Gibraltar over a multi-month timescale, with 82% showing signs of life. That's certainly a good result. 2, that morphological regression is not the same as death. So I think that's also a very key finding. Rhizome development, we've been able to show that is achievable under nursery conditions, and each conditioned plant has a very important conservation value within a nursery environment. So those are the conclusions that we've just seen under the phase 1. Our priorities going forward, we want to investigate the specific conditions in system A to identify what exactly was different, why plants there seem to do well or better than in other ones, because it could tell us something about the nursery environment that is important. To build the recovery protocol and those recovery tanks is something that we're going to be looking at in the future, and having some controlled experiments, varying the light, varying the conditions, in order to see how the plants are reacting under different treatments. And what we want to try and drive towards is seed collection, because in my view, getting seedlings into the wild is probably a better approach than getting the established plants that we have and moving them into a condition where all those variables that keep them alive are changing again. So at least with seedlings, what we're doing is we're taking, if you like, the profit that the thing has made and moving those out into the environment. So those are the options that we have. So thank you very much.
Right, and then we have our last talk. We have from Ainhoa Idiondo Molina. Ainhoa is a PhD student in marine science and intelligent marine robotics, with a focus on AI-assisted ecological monitoring in marine environment. It explores how smart observatories and marine robots can transform water observations into ecological indicators.
Hello, good afternoon. My name is Ainhoa Idiondo, and I'm going to present the project I foresee and how we can bridge traditional methods and robotic-based approaches for seagrass monitoring. The main object of the project I foresee is to develop a system that uses underwater robotics and artificial intelligence for the mapping, restoration, and monitorization of the Posidonia oceanica. This project involves 4 different partners, where each one is responsible for a different part of the system. The University of Valors is responsible for the habitat mapping and developing methodologies for extracting important characteristics of the plant. The University of Girona is responsible of developing methodologies for the intervention and restoration part, such as developing a robotic arm to plant Posidonia. And the UPC is responsible for developing methodologies for environmental monitoring and also detection of biodiversity in the area surrounding the Posidonia. And lastly, the ICM is responsible for the ecological assessment of the Posidonia, the ecosystem. Okay, so the current problem with marine observation is that they are local, temporally spaced, and costly due to personnel and material needed. And that's why the main objective of the ICM and my thesis is to develop methodologies assisted by AI to extract ecological indicators from data acquired by robots and environmental sensors. Also to develop an ecological model with AI to detect stress or anomalies in the Posidonia and assess the habitat conditioning, as well as predict the evolution of the system. Also to support spatial upscaling to find where local observations can be transferred to a larger area. And lastly, to integrate all this data and models to a digital twin simulator for monitoring and decision support. Okay, so at the moment, Posidonia monitoring is relied mainly on data acquired manually by divers, and they extract information such as the plant condition or the structure of the meadow. This information is then used to calculate ecological indicators. And these indicators follow the Marine Strategy Framework Directive that uses indicators from different ecological levels to assess the health of the ecosystem. So here we have some different indices that are used to calculate the health of the Posidonia, and we have 3 different types of indicators: the structure, morphology, and physiology. For the structure, one example would be the number of shoots in 1 square meter, or the deepest limit of the meadow. For the morphology will be the length or the width. And for the physiology, it's, for example, the nutrient content in tissues, that this one requires a laboratory analysis posterior to the diverse measurements. So the idea is to adapt these ecological indicators so that a robotic system can measure them. But we have some problems because, for example, the physiology ones, we cannot make them, well, we cannot adapt them for being done by robots, and some of the morphology ones, they require a very precise measurement, therefore we cannot do it exactly as a diver would do it. That's why the AUVs can do bathymetries, photomosaics, as well as collect environmental data, and then we can adapt these indicators and put them in ecological models to assess the habitat. This doesn't work if we don't have a validation framework. The idea is that when the robot goes takes the different measurements, also for our divers to take the same measurements, so we can compare them and have a ground truth. One example would be the shoot density estimation. As I said, a diver has to count the shoots. Usually it's in a 40 by 40, and then they extrapolate to 1 square meter. So the robot cannot count the number of shoots because it's really precise and also really smooth. So we cannot do it by computer vision. But the idea is that maybe we could do it with detecting the meadow cover or agrograph leaves, and then extrapolate the information and compare them to see if we could find some proxy. As I mentioned before, we can evaluate the the Posidonia by looking at different information domains. The first one is the landscape of the habitat structure. This one will be like the middle cover if there's some fragmentation. The second one will be the morphology. This one focuses on the plant individual, such as the length or the width. As I said, the physiological status, we cannot do it without laboratory analysis. So it's out of the picture. Then the habitat conditional or environmental drivers, such as the temperature, salinity, the light availability, and lastly, the biodiversity and ecosystem composition, detecting fauna associated with the Posidonia or some changes in the community. With all this information from these domains, we got a clearer picture on the health of the ecosystem. Okay, so this is the architecture overview of the project I foresee. So we need a lot of systems to make all this part works. First of all, we have some historical information such as satellite data and also historical data sets that give us a context of the Posidonia until now, how it's been developing. And then we have some different AUVs. There's one chart of the mapping or the intervention, also the fixed observatory and sea landers. That this one will be the ones to measure all the environmental variables. Then all this information will be put into an ecological model who will assess the health and predict their evolution, and will put it all in a digital twin for decision support. Well, the first thing that will happen will be that an AUV will map the seafloor of where the Posidonia is, and with the AI models, we extract the information of the important characteristics of the plant, and then all this information with the data from the environmental sensors will be applied to the ecological modeling, and after the ecological modeling and the prediction, with this information will help us to see if there is some place where we need to do restoration. So the industry in this case could go and plant a new Posidonia. Basically, all this is based on a long-term monitoring. So to make different surveys from like 2 to 3 years, and we'll integrate it with the digital twin, and of course the validation of all the system. Okay, for doing this different scientific requirements, we will need some platforms. Here's some examples. To map the Posidonia, we'll need to do bathymetry and photomosaics. We will need an AUV. Also, we will need the operational requirements, such as the survey area, that this will be around the UPC. The frequency, it will be kind of every half a year or a year. Okay, the expected results is to identify areas where local observations can be extrapolated to a wider area or regions, and have these adapted ecological indicators, as well as the validation framework to compare the measure extracted with the diverse measurements. and lastly, have tool to support decision. Well, that's it. Thank you very much.
Thank you very much.
We're finished. We've got still a bit of time left to answer questions.
We have two new questions for Evgenia. I'm not sure if she's still with us and she's able to answer, because I can see her online, but I'm not sure if she's able.
Yes, I am, but I have 5 minutes. I need to leave, so yes.
Okay, thank you. Yeah, there are questions.
What are the conditions regarding the carrots? Well, as I said, what do you mean? The numbers, the actual numbers. Who has asked this question? So,
it is written, so probably.
Yeah, sorry, it was me. It's also online, so maybe I could ask in person. It's easier. I was wondering what are the conditions regarding the flow of the bay, and if it's a bay that has a high interactions with bays nearby, because I don't know where the location was. Thank you. Sorry.
No, it's okay. Yes, so I don't remember really the numbers now. I can look it up and you can email me and I can send you the numbers. But no, it's a typical bay of Greece. Is semi-exposed. Also, I mean, the part where I show you where it's mostly the dead mud, it's more sheltered. The important thing is that that what differentiates, let's say, the stations is that the ones that are located in the inner part of the bay are closer to the fish farm and also in the direction of the main water current. So basically, they received the effluence from the fish farm to a higher extent than more exposed, and they are more sheltered as the geomorphology of the sites. So that's why they accumulate more silty sediments also. Regarding the EPQI, the results are not published yet. I'm trying to finish this paper, so they will be out soon. Depths are collected, the standardized indicators of Posidonia work at 15 meters. So the samples are collected in the 50 meters, but also in the upper limit, because this is how this indicators works. And also the lower limit has to be measured. And how many were taken? How many were taken of what? For the blue carbon. The blue carbon. So the blue sorry, I replied for the seagrass. So the blue carbon, the stations in the sheltered part of the bay were between 6 and 8 meters, and in the exposed part there were 12 meters depth, which was the shallowest high cover place that we could find to be compatible. But based on the assessments that we have done in Greece, in a lot of stations in the South Aegean, we know that the threshold is below the 15 meters, where you see change in the both in seagrass production and also blue carbon. And then, 3 replicates per station.
Okay. Thank you, Evgenia. Thank you very much.
I have to go. Sorry, bye. And thank you again.
Thank you once again. Okay, Frederick, do you want to start with the first?
Yeah, sure. A question for Gigi. I don't know if you said it during the presentation, but in which period of time did you achieve the 85% of survival rate in your experiment?
After 3 years.
After 3 years.
Yeah. So obviously most of the research projects don't start talking of actual survivorship in Posidonia after 3 years. So we had the results. The representative was of 2.5 years. The data, I wasn't able to upload it, but the data from 3 weeks ago is slightly lower. But because also, so where we did the replanting, it was completely signaled, no anchoring. But obviously people do whatever they want.
Yeah.
And they did destroy 2 of our plots.
Oh, shoot.
So that brought the survival rate down. We still found other methods. Actually, a lot of the fragments stay rooted without the attachments, which was actually really nice because the aim of the anchoring techniques is just to give the time for the roots to grow and then stay by themselves.
Right. Another question?
Our rhizomes, no, they're actually next to where we're going to be replanting. So we take fragments that have been broken off from the local prairie either by storms or by bad anchoring techniques. We dive and we get them just near the areas where we're going to be doing the replanting, which, quick tip for those of you guys looking for fragments, in the prairies that is a bit patchy, they collect in a bit of the deeper side, like in between the prairie where there's no actual seagrass. They just kind of collect over there.
I'm going to sneak in an extra bit then.
Yeah.
Your community outreach people, the people who are doing it, are they diving or are they snorkeling?
That's actually another of the questions there. Are you on this? No, actually, so we have, as we did mention, we have different, the way we do the monitoring and the replanting is we try to involve divers and non-divers, so there's actually a shore for everybody. Obviously, the fragment recollection is done by divers, but when we do the bigger planting sessions, we have got people on the shoreline that are doing the measurements that are not divers, and then people that are comfortable with snorkeling. They will sometimes be relaying the bags with the collected fragments over to the people on the shoreline so that they can start counting, measuring, and then we also label each fragment is labeled individually, so we can actually guarantee a follow-up of each fragment independently.
Okay.
So you answered both? You want?
Yeah, and why do you consider that frequency in order? So we did, before we started our research, before we started the project, we grabbed all the review that was available up to 2022. The project started in 2023, and we looked at the periodicity that had been established in previous projects, and we just kind of, why are we going to reinvent things that have worked up to now? So we did what had been done up to the moment.
Okay. Thank you. For Paul, I would like to know how you designed these study stations and why you decided to study that area, and how did you design it then?
Paul's.
Oh, Paul's not here.
No.
Okay. Sorry. For Lewis, how do you achieve this accurate and individual monitoring for each Posidonia plant in the tanks?
So each of the tanks has 5 plants, and they are positioned strategically within the tank, like on a 5-sided die in that type of structure. So each plant in that position has a particular label that's not actually in the tank, but we know based on the position that's there. Because it's a tank, what we can do is we can easily measure leaf length, we can measure the number of leaves that are there, we can measure the length of the rhizome, and look at the roots as well. We use a lot of photography in order to see the development of the plants over time. So whether that's, with the word accurate, if that's accurate enough, I'm not sure, but that's the sampling strategy that we use for the tanks in the plants in the tanks.
Thank you. Those are the questions from online. Does anyone have any additional questions?
I do. I have a question for Alex. You talked about a viability of leaf count of 7 leaves per fragment. Why? And what do you mean, like 7 leaves per fragment in total, independent of how many shoots they originally had?
Yeah, exactly.
How do you guys get to that being your break point of the viability?
Well, we counted the number of leaves once at the beginning of the experiment after 7 months, and we've seen that the plant that has more than 7 leaves had a higher chance survival because all the plants that had like around 3 or 4 leaves after 7 months were dead in the sea.
And you talk about leaves or shoots?
About the death of the.
No, yeah. So you mean like actual like way you guys selected the fragments originally? Did you have like a minimum length for each fragment and a minimum number of shoots for each fragment?
No, we take every fragment that we find in the leaves, and then we land it to the sea, and we see that the fragments that had around 3 or 4 leaves died after 7 months.
Okay.
Any more questions?
Just a quick comment for Lewis. Your plants came from Malta. You just kind of in that genetic east-west convergence. Is there a possibility that there might have been a genetic component to your spontaneous leaf formation, for example?
The truth is, I don't know. It's certainly a possibility. Yeah. We actually did try to source plants closer to home, but it was the emissions and protection are such in order to so with also it was easy.
Jeffrey? No, Leon?
I have another question for you. Since you're treating with 2 different administrations, 2 different states in the bay, and there are multiple industry pressures, oil, metals, whatever. What is the habitat suitability projection, let's say for 10, 20 years from now? Sorry.
Well, I think so I can't answer that question because we haven't done that work. I mean that's straight up. But I think now that the grasses are in situ and because we've got industry players that are paying for it, these questions are now starting to become more important to them. It makes it more relevant and that'll help in improving that habitat suitability going forward. They're investing money fundamentally in a seagrass project that they need to see go out into the environment, right? And it's that agency that is important in order to improve the
Thank you. I think that's it, no? Well, thank you very much, everyone.
Thank you.
That's for sure. Thank you, Lord, for me. I would like to thank Marcia, Geltrú, and Simonetta from the University of Naples because they help us a lot for the preparation of the agenda. And of course, Joaquin, Frederick, Ingram for organizing the event. And all of the other partners, Jacobo and Vesna, thank you.
Thank you, Elena, for a wonderful.
Thank you.